Nicobar Island for conservation of traditional land races of rice. He also got ICR Fakhruddin Ali Ahmad Award for outstanding research in tribal farming system research in 2018. He is a recipient of fellow awards of National Academy of Biological Sciences, Indian Society of Genetics and Plant Breeding, and Indian Society of Coastal Agriculture Research. I hope with vast experience and knowledge what he has acquired throughout the years, this lecture will be more beneficial to all of you. So, sir, with this, I would like to welcome you here.
Thank you. Shall I share my screen?
Sure, sir, sure.
So continue double click in presentation view mode.
Yes. Thank you. Thank you, Dr. Kuldeep, for the kind words.
So slideshow mein nahi hai, sir. It's not coming in slideshow.
Okay.
And first slide.
Slideshow. Is it now on slideshow?
No. एक बार अनशेयर करके शेयर करिए. First open your PPT, then share slides. ये वाली now it's okay. Perfect.
It's okay now. It's in slide
show. Yes.
Okay, fine. Thank you very much, Dr. Kuldeep Tripathi, for the kind words. And I also welcome all the trainees to this training program. And to start with, I would be sharing our knowledge with all of you on principles of germplasm characterization and evaluation. My lecture has today two components. One is on characterization and evaluation principles. And second, I would like to give you a brief overview of our division of germplasm evaluation, what kind of activities we are doing at ICR and BPGR. I'll try to share all the information with you during this lecture. Well, the goal of PGR management, I would like to share with you. And the principal objectives of plant genetic resource management are to augment, regenerate, maintain, distribute, preserve, characterize, evaluate, and utilize the genetic diversity. Since you all are early career scientists, almost most of you are early career scientists or faculty members. So I would like to share our knowledge on augmentation. Augmentation, the word indicates that you need to enhance the your germplasm strength, maybe through your explorations, collections, whatever it is. Regenerate. As you know, the viability of the material of the seed material is also very important. So and it depends from crop to crop, and we need to regenerate the material from time to time so as to ensure the viability of the material. Maintain. How we maintain this germplasm, because we have more than 4 lakh germplasm accessions in our bureau. So you can imagine how much work is involved to maintain the all database and whatever it is. Distribute. It's not only for archival purpose, but unless we do not share the unique germplasm or whatever germplasm required by you, it's of little value. Preserve. It's very important to conserve or preserve the material for long-term use, and for that we have state-of-the-art facility in our bureau to preserve. Most of you may also be having this facility like medium-term storage and long-term storage in your institutions. Characterize. Then unless we do not characterize our genetic resources, they are of less value. And evaluation is also important to reap the benefits in economic terms. Utilize the genetic diversity, and how the plant breeders can utilize the genetic, these resources. It's also very important. And PGR management should pursue all these goals rather than simply acquiring and storing accessions. As I already mentioned, that merely storing and preserving will not be of any use unless we do not utilize these genetic resources. The outline of the basic terms is like germplasm. It refers to the sum total of all the diversity existing in a species. Characterization is description and identification of an accession. Evaluation is how much economic worth a germplasm has. So it comes through evaluation, proper evaluation through scientific principles. Plant genetic resources refer to germplasm or genetic diversity of actual or potential value. Unless we do not have any economic or significant importance of the genetic resources, we may not call it really plant genetic resources. So they must have something unique, and the uniqueness varies from time to time. Today something maybe may not be useful, but after maybe 100 years it may be of a significant value. So almost all germplasm are we call plant genetic resources, and the genetic resources, they are identified through germplasm evaluation and characterization, and further their use in the crop improvement programs. So definition of the characterization and evaluation, I think you already you are clear about these terms. The characterization is assessment of attributes in a given accession that are relatively stable across environments. Mostly these are followed for the qualitative traits, and evaluation is mostly for your quantitative traits to make it more simpler. And these attributes are almost as synonyms of traits or characteristics. So characterization and preliminary evaluation, the objectives encompass, describe an accession, classify group of accession based on sound criteria, allocate specific and unique traits, estimate genetic diversity of the collection, recognize the duplicates. Yes, this is very important, because there may be many collections which are genetically similar with each other, but in our database or in your records, they may be separate. But we need to really differentiate these duplicates from each other, and they can discard the the duplicates and keep only the real ones with you. Estimate extent of variation in the collection. Unless we do not understand and utilize the variation, we cannot come out with the useful variability from our material. Then supplying germplasm information to the users. As I mentioned, that unless we do not share the unique material with each other, it's of little value as far as the welfare of the humanity on this planet is concerned. There are different nowadays mechanisms, different memorandum of understanding nationally as well as internationally, how we can regulate this sharing and supply of germplasm. Develop a core, mini core collection and reference sets. So I will not go in detail on this topic, because we have experts already. I think Dr. H.D. Upadhyay covered this topic yesterday. So these are the general guidelines for characterization and evaluation. What kind of material or species we are going to characterize, it is very important. What type of collection it is, whether it is a base or active collection, when to characterize. So we need to decide and plan carefully in which season we need to characterize the material so as to meet the objectives. What should be the sample size? The total population size has to be planned and executed accordingly. Site for characterization. So preferably our site for characterization should be similar to the growing conditions of that crop. wherein the idea is wherein the maximum expression of the material is manifested. So we need to select this site very carefully and wisely, and it should be preferably free from diseases, pests, and other abiotic stresses, so that the material which is sensitive to those conditions is not killed in those conditions, and we may not be able to collect data from such collections. Materials for characterization, and in this new collections are obtained through explorations, as I mentioned, and our bureau is regularly conducting this activity to explore and collect material from diverse locations in our country through our regional stations, new exotic introductions. When we feel that we have a little variability in our indigenous material, we import the material from the different countries, new accession generated from conventional and biotechnological methods, samples from other gene banks, and then characterization techniques, on what basis we are performing characterization, what are the traits. So the traits are agromorphological traits which are visible to us, chemical, biochemical markers. These are another specific traits through which we can differentiate our accessions, and molecular techniques, DNA markers, and nowadays the novel DNA markers, they have come up like SNPs and whatever. So they are employed to uncover the variability in the material with very high resolution. So agromorphological characterization, as in a simple terms, whatever we are able to see in the field. So based upon those traits, the agromorphological characterization is performed. Biochemical characterization is based upon the biochemical traits, like your abscisic acid, protein, it's all the sugars. So all this characterization is performed in the material so as to uncover the unique traits in our germplasm, which are biochemically superior. Molecular characterization is nowadays more popularly being used, and they have very high resolution. And whatever is not resolved phenotypically, so molecular characterization is able to perform that role. And diversity for one character, I would like to say, the diversity of one character may not be equal to that for another character in the same germplasm pool. Suppose you have 500 accessions of any crop. So you are looking for any trait like disease resistance to a particular disease. You may not find variability for disease resistance, but if you screen the same set of material for another trait, you may find substantial variability. As we know that these are different soybean accessions, and you can see different colors of these seeds. So they are obviously, as you are able to see, diverse looking, different from each other, but they may be susceptible to any disease. So as I mentioned, that diversity for one trait may not be so for another trait. So we need to screen our material, the same set of material for different traits, so as to find out the worth for that specific trait. The components of characterization and preliminary evaluation include multiplication, descriptor list, defined traits, and measurement data. We have sometimes a little quantity of the seed in our gene banks. So first we need to multiply the seed so as to conduct the experiment in the field. Descriptor list, we need to decide what kind of traits we are going to record on that material during characterization. And nowadays we have descriptor already available in different crops. I will share this thing with you in succeeding slides. And measurement of data, these are mostly related to the quantitative traits. Yes, so these are different scales followed for recording different traits, like interval where you have 1 to 5, then 5 to 10, then 11 to 15, something like that. And these interval scale, this interval scale is mostly followed for quantitative traits like your height, lead components, ratio. So ratio, as you know, you may be you are knowing how we calculate the harvest index. It is the wheat. If we say it is grain yield divided by biological yield into 100, so that is a ratio. So that way we calculate the harvest index percentage, ordinal value. So mostly in case of biotic and abiotic stress, this scale is followed, 1 to 9 scale. 1 means highly tolerant and 9 is highly susceptible. And then we have mostly these are the odd numbers actually, 1, 3, 5, 7, 9. So these are called ordinal scales. Then nominals are qualitative traits. These are easily visible to us, like color, seed pattern, growth habit. As I showed you, the soybean seed colors. So those are the nominal scales. Binary is like presence or absence of a particular feature in the germplasm collection. If the pink color is present in our germplasm, in our any collection, it will be yes for pink, and if it is other color, then it will be no, 0 or 1. This is the view of our large scale mega characterization of our entire collection of lentil at Isapur farm. You can see how many efforts are required actually to conduct these characterization experiments, and but it's not that difficult. Also, if we have we have intentions, planning, and execution plan, we can do all these things in a good manner. So the variability of seed characteristics in rice bean, you can see this is for seed characters, and similarly we have the diversity for numerous traits, which are required by breeders in different locations. The characterization and preliminary evaluation, this also includes the descriptors and descriptor states. And descriptors are something, in a simple term, I would like to share with you. The descriptors is any trait which identifies or differentiates an accession or germplasm collection from another, collection. Like you have color, pink color, or any flower color. So based upon that, you are able to classify the different germplasm collections. So we can call color is a descriptor trait. And internationally and nationally, there are descriptors designated by different organizations. Like we have IPGRI, International Plant Genetic Resource Institute, and then NBPGR. So these are the world-class institutes which have developed these descriptor traits for different crops, and these are employed for characterization of the germplasm accessions. As I mentioned, these are the descriptors designed and compiled for different crops by IPGRI, an international organization. So these are the known documents. Similarly, ICR and BPGR has also developed the descriptors and published these documents for different agri-horticulture crops. Crop descriptors, these provide an international format and a universally understood language for plant genetic resources data. These are also targeted at farmers, curators, breeders, scientists, and users. So these are a little bit scientific, but then scientific traits. International multi-crop passport descriptors. So these were published in 2001, and these multi-crop passport descriptors are widely used as the international standard to facilitate germplasm passport information for this purpose. descriptors for farmers' knowledge of plants, because farmers are not aware of the technical terms, so they also may be having their own traits based upon their traditional knowledge. So they are also amalgamated with our scientific descriptors to really differentiate the accessions. So these are descriptors for farmers' knowledge, which could be easily understood by farmers. So what are the principles while the principles followed during characterization and evaluation? These should be recorded in the CGS scale. Presence and absence of characters are scored as 1 and 1, as I mentioned in binary scale. Many quantitative descriptors which are continuously variable are recorded on 0 to 9 scale. Qualitative yes, for most of the qualitative traits, the last state is others. Suppose you have descriptor list, 30 traits are there, and within those, suppose color, you have already documented 5 traits which are enlisted in that list. But if you find something extra which is not in the list, so that comes under others category. So the severity or extent of infection or infestation may be recorded on 1 to 9 scale. Suppose you are recording data for drought tolerance. So what scale you will follow? So there is a popular nowadays, you may be knowing, that stress susceptibility index. So which is stress susceptibility index is based on the amount of reduction experienced under stress compared to the non-stress conditions. So that is also followed for evaluating the materials. A standard color chart designed by Royal Horticulture Society color charts or computer simulated color charts are very useful to determine the color of the flower or other plant parts with high resolution and with more scientific value. The detailed evaluation is mostly performed for agronomic traits. Agronomic traits I mean to say your traits of economic importance, like seed yield, grain yield, and then abiotic stresses, like drought, salinity, submergence. So evaluation is done for these abiotic stresses also. Biotic stresses include your diseases, pests, weeds, whatever it is related to biotic problems. And the quality, mostly it is performed through biochemical estimation. And so that way evaluation is done for all these separate category of traits. Evaluation for biotic stresses, this includes preliminary screening, field evaluation, advanced evaluation. Now I would like to share with you that if we talk about the biotic stresses, so unless we do not thoroughly and rigorously test our material for any biotic stress, it's not going to be sustainable, because you may be knowing that the pathogens or these pests, they keep on evolving, and we must have the strong resistance in our germplasm, preferably conditioned by more than one gene, because the pathogen mutates at different loci and then overcomes that resistance. So we should have very strong resistance confirmed over years across locations. So first we start with the large number of, say, 500 or 1000 germplasm in your location for a screening for any disease. Then you come out with a few lines. Suppose from 500 you come out with 50 lines which seem to be resistant during first year. Then you evaluate them during second year, preferably in the replicated design. And then your data is, because you have a reduced number of genotypes and you have you can do it with more precision. So these are the data on disease severity or incidence is collected from 3 replications or 2 replications. Then you come out with, say, from 50, you come out with 10 lines or maybe 5 lines which are showing good resistance. Then these 5 lines should be evaluated at multi locations. So as to, idea is that your multi locations where that crop is popular. So idea is to expose your resistant material to different growing conditions, because then you expect different pathotypes in different locations. So if your material is say resistant at 3 locations, it is expected that it is having resistance against many pathotypes or the isolates. And then suppose from 5 you come out with the 3, then these have to be artificial. These have to be tested for disease resistance under controlled condition, maybe through artificial inoculation. And these should also be exposed at hotspot location where we have these disease conditions. This is a view of mung bean, and it was tested against mung bean yellow mosaic virus under field and controlled conditions. So this work was done in our division, and initially around 340 sessions were tested for this virus condition for this virus under field conditions. And this from 344, 8 putatively resistant germplasm were found, and then these were further confirmed through artificial inoculation, and these accessions finally proved to be resistant to mung bean yellow mosaic virus. Similarly, this is a kind of a case study in case of soybean. What we did is soybean is normally adapted to kharif conditions, especially in North India, you may be knowing. But we were interested to have the kind of material which is surviving or performing well during the spring season. That means sowing of soybean is done in February, and then during the flowering initiation, as you know, it coincides the months of May or June. And then if you grow soybean normally in the Rabi season or hot summer in North India, so most of the accessions, they keep on growing without setting seeds. But we were interested to know if there is any variability in our germplasm. So we exposed our entire gene bank accessions numbering around 8,000, and then we did this evaluation work, characterization firstly, and then evaluation, and we could find that we could find around 42 accessions from 8,000, which were 39 accessions from 8,000, which were showing very early maturity or early maturity compared to the best checks. So that way, from 8,000, we came to initially 250 accessions, which were putatively early. And from 250, we finally came in next season, 39 accessions, which were showing early maturity. So this way, we are able to narrow down our accessions to meet our objective for selecting for a particular trait like early maturity. So this is a view of early maturing lines in soybean. So we were looking for the early maturing lines. These are the checks in the background, which are not yet flowered or set seeds, but we got the accessions which showed a distinct early maturity. As far as evaluation for quality parameters is concerned, nowadays in the era of this malnutrition and hidden hunger, these biochemical traits are of significant value if we are to free our this suffering population from these malnutrition and other deficiencies. So in wheat, the important traits are in protein, gluten, sedimentation value, iron and zinc. In maize, protein, tryptophan, starch, oil, fatty acids. And now we have germplasm, which are having all these superiority for these biochemical traits. In oil seeds, it is primarily the oil content, fatty acid profile, anti-nutritional factors, and the erucic acid is also a very important trait. Pulses, protein, amino acids profile, oligosaccharides, phytates, sugar. Potential crops, which are underutilized, we also call them underutilized crops. In them, protein, sugar, starch are the important factors. But in vegetables, we prefer the sections which have better minerals, vitamin C, sugar, starch, phenols, carotenoids, especially like a crop in carrot and other yellow colored crops. In medicinal and aromatic plants, we look for the active ingredients, essential oil, and the chemotypes, which have specific chemical compound in a large quantity. We look for such traits in our medicinal and aromatic plants. So in our division of germplasm evaluation, we have a very strong group on biochemical this work. So multi-location evaluation in important crops, and it is normally conducted in sync with the eclipse of respective crops, like say wheat. It is eclipse in Karnal, Indian Institute of Wheat and Barley Research. For rice, it is Indian Institute of Rice Research in Hyderabad, and similarly for almost all crops we have this eclipse system. So it should be done preferably in collaboration with respective this directorate or eclipse system. And parameters include agronomic traits, biotic, abiotic stresses, quality parameters. Yes, I would like to, though we have separate lecture, many lectures on molecular aspects as far as their their relevance and utilization application in case of germplasmic sections is concerned. Just I would like to say that these are nowadays the novel tools to uncover the hidden variability in our material. Sometimes you have like 10 accessions, they're looking phenotypically similar, but they have significant diversity from molecular points of view. And these are mostly utilized now different classes of markers. Now we have more precise markers which could be employed for phylogenetic relationship understanding, development of core collection, gene flow study. Yes, DNA fingerprinting is very important. And now there are now the government guidelines which we are following, and any variety which is proposed to be released has to be DNA fingerprinted, so that nobody else can claim that this is my variety or somebody else's variety. So the record is very useful to ensure the IPR issues as far as the varieties are concerned. The gene discovery, yes. So this is also very important. Sometimes you are not knowing whether the drought, how to really screen the material for drought tolerance. But if you have a gene which is already reported is conferring drought tolerance, then you can screen your entire set of material, entire set of collections against that screen for the presence of that DNA marker in that collection, and you can find some collection which possesses that gene, and that accession may not be required for feed evaluation. So that way gene discovery is also done. And trait discovery, trait specific germplasm is also screened through these molecular markers. the core collection for evaluation and core collection, though there was a lecture yesterday, I would like to just only say that core collection refers to a representative sample from a big population, which represents the maximum diversity existing in that population. The advantage of course is that you can easily handle the few collections compared to the large population size, and that particular your population with the reduced size can be employed for all kinds of genetic or the other evaluation works. Core collection may be developed by using information on accessions of collections, including passport data. So based upon these traits, the cores are developed: passport data, characterization data, evaluation data, biochemical traits, molecular traits. A combination also can also work. So these traits singly or in combination may be employed to develop a core in a particular crop. So our bureau has developed core sets for crops like sesame, brinjal, okra, wild lentil, mung bean, wheat, and chickpea. And we are in the process of further developing cores in other remaining crops also. So this is an example on large scale characterization of wheat accessions, and there is a world record for this germplasm characterization work, which has been published and documented in Limca Book of Record also, that in one go and in one location 22,000 wheat accessions which are conserved in our gene bank were evaluated, characterized in simultaneously. So this was a massive task, and this is a success story, and it led to many many significant findings, and still we are working on this the mini course or the course developed from this characterization work. It was published in very high peer-reviewed journal also, this work. So these are the I wanted to share this information with you on the large scale characterization work done by our bureau. Similarly, in chickpea, based upon different traits, around 1100 accessions were derived from 1400 accessions conserved in gene bank. And documentation of information is also very important. So we have a very strong group on the digitization of this all the accessions, and so far NBPGR has characterized more than 1,81,000 accessions, and these are published in the form of catalogs across almost all crops, all field and horticulture crops. Registration of genetic stocks is very important, and you may also many of you might have also planned for registration. Some of you may also have registered your unique germplasm with ICR and NBPGR. The idea is to submit unique material in our national database, and we have a separate lecture on this very important activity. What are the guidelines? What are the principles through which you can, register your material with our bureau? So it will be going for long-term conservation and record in our national system. These are the unique accessions which have been identified during the course of our this characterization work. And like in your bottle gourd, these are the kinds of different leaf types which we identified. In view of vitamin A deficiency, orange fleshed cucumber is also identified. We have furrowed a fruit in bottle gourd, a different kind of fruit shape or color. And similarly, such unique germplasm in cucumber also identified. The unique germplasm identified through characterization and evaluation in Indian mustard has also been reported and documented, like broom type siliqua orientation, this accession, and then we have middle branching erect type of this accession, and then lacerate type of leaves. This is a different kind of leaf. These are different kind of leaves compared to other leaves. Then a different kind of siliqua also. So these are the different trait specific germplasm registered by our plant genetic registration committee. And in wheat, these are the different germplasm which have been registered. Similarly, the germplasm in mustard, black gram, and cucumber have been registered. Like in chickpea, you have this kind of upright podding, which is not normally found in the normal germplasm. For millet, you have a high popping yield, this collection. Similarly, in this crop, linseed. So we have identified and registered different kind of accessions in various crops. Yes, this is about the germplasm field days, which we conduct from time to time. And our division of germplasm evaluation is mainly responsible for this work. And what we do is we facilitate our NARS, mostly the breeders, for a particular crop in selecting the germplasm suited to their respective conditions. And we conduct the characterization and evaluation work for a large number of collections in that particular crop at our farms. We have around 100-acre farm at Isapur near Delhi, and 100 acre area in Isapur, and around 10-acre area in ICR NVPGR Pusa farm, where we conduct the large scale characterization and evaluation, and we invite the nearest breeders to see the material and select the material of their choice. So utilization of germplasm evaluated is also very important. Unless the material is not utilized by breeders for developing varieties, this is of little value. So we have many. We are compiling the data and documenting this information. How many accessions facilitated by NBPGR have been converted into the varieties? So these are the examples of some of the accessions which have been developed into varieties, and they are serving the cause of food and nutritional security of our country, and maybe some of them have been released as varieties in other countries also. So nowadays, with the availability of digital information science, we employ these digital resources to record data and to digitalize our information on plant genetic resources. And I hope there is a separate lecture on this aspect, how we utilize these digital tools for recording, maintaining, and utilizing the information on the plant genetic resources. Nowadays, as you know, phenotyping is becoming more precise and high throughput, because it's not easy. You can see in the background, thousands of these accessions being characterized at one location, and you can imagine the amount of work involved to record the data on different traits. But nowadays we have the availability of UAV based phenotyping or drone based phenotyping, through which you can record the data in very less time, and the data will be very precise compared to your manual recording. So recently we have initiated this, we have made the initiative for this approach, and we are following nowadays this approach to record data for many traits in our large number of collections being characterized and evaluated. So similarly, we have the UAV based phenotyping for high throughput screening against drought stress, and with the very less time we can have more precise data. And this is now we must use these modern tools to characterize, evaluate, and phenotype our material. And this is going to reduce the drudgery and manpower for recording data. What are the lessons to be learned during characterization and evaluation? So complete characterization and evaluation of accessions leads to trait discovery and ensures the effective utilization of the germplasm collection. It's not only for academic purpose, but if you are characterizing and evaluating the material with your defined objectives and clarity of goal, then you are certainly going to get unique material which could be utilized for developing varieties. Understanding of the taxonomy, agromorphological, and genetics of the crop species is essential. So you have suppose 5,000 accessions in your gene bank, but unless it is not grown, you are not able to classify it taxonomically or delineate it taxonomically. So it's very important to grow them under field conditions. You may find some unique accession also, unless you do what I mean to say, unless you do not grow, you may not be knowing the uniqueness of that material. Characterization and evaluation are indispensable to estimate the extent of diversity. Suppose we have 5,000 accessions in our gene bank, but if we do not characterize them, we are not we may not conclude whether the diversity desired diversity exists in that material or not. Another thing is, while you are doing characterization and evaluation, you are also multiplying seed of those accessions, and those seeds, because we have limited seeds normally in our long-term storage or medium-term storage facilities. So seed derived through characterization and evaluation can also be deposited back to our gene banks so as to augment their number. The strategies for characterization and evaluation, what should be the ideal conditions actually, and for this characterization and evaluation? So we need to decide and finalize the population size of our collection, which is being targeted for that characterization or evaluation, so that there is there is not any kind of somebody please can
Please mute.
please mute. So frequency of multiplication and regeneration cycles should be as low as possible to minimize the loss in genetic diversity. The characterization should be done in non-stress environment corresponding to the collection site. As I earlier mentioned, that characterization of your collection should be done and preferably under non-stress condition, wherein you do not, your material your maximum material is survived, so that you can record data on maximum number of traits. And appropriate techniques should be practiced depending upon types of breeding behavior and biological status. And we need to, because while handling this large number of collections, you need to be careful so that you do not contaminate one accession and you do not mix one accession with another. So you need to do it very carefully and properly. So I have completed my talk on principles of germplasm characterization and evaluation. Now I would give you an overview of activities being performed in our division of germplasm evaluation, just an overview and for your information. See, the major objective of our division is characterization, evaluation, and documentation of genetic resources of agri-horticulture crops and their wild relatives for their efficient management and utilization. See, it's not only the cultivated types which are important for us, but the wild relatives are equally or even more in more value as far as their utilization is concerned. And you can see the large scale characterization work being performed at our NBPR ISA farm. The major activities of our division is these include characterization and preliminary evaluation of major agri-horticulture crops for important agromorphological descriptors. Then our objective is also to develop cores for different crops, not only cores but reference sets also. So reference sets are normally these refer to the any trait specific population from the germplasm collection set. So suppose you have a resistance in from the total population, your germplasm collection is 1000, and you have YMD resistance in only 100 collections. So you call that 100 collections are your reference set for YMD in that particular crop. Evaluation and identification of trait-specific germplasm for agronomic traits, biotic and abiotic stresses, including quality parameters. Pre-breeding, as I mentioned, is very important, and we have nowadays this wild garden established at our farm, wherein we are growing and displaying the different wild species of different crops. They are very important resources, not only for today but for future also. And another activity, as I mentioned, is important, that is germplasm field days and germplasm supply. We are also supplying seed as per the demand of different breeders from different institutions in India, so as to facilitate them not only for varieties development but also for conducting basic studies. So these are the infrastructure facilities available in our division. We have around 10-acre area in NBPR Pusa farm, and Isapu farm we have around 100 acre. Biotic and abiotic stresses, these are being the work on these stresses is being done in net houses, rain out shelters, and we have very strong group of people who are working on biochemical aspects, and we have GCMS, NIRS, AAS nitrogen spectrophotometer, and many other state-of-the-art equipments in our lab. And another significant addition to our facilities has been on the field gene bank of perennial crops, the horticulture crops, and this facility has been inaugurated last year by Honorable Director General ICR. So these are the different kinds of infrastructure facilities in our division. This is an example of 1 year germplasm characterization work done by our scientists in division of germplasm evaluation. So we have characterized and evaluated more than 20,000 accessions in 1 year across different crops. So now next is on core sets development. we not only teaching, the concepts of course, but we are actually following the concept in developing course in different crops. Like in rice bean, we have 250 accessions developed as core. In linseed, 2657 accessions for core. In mung bean, we have 252 accessions developed as core, and these were derived initially from 4,000 germplasm collection. You can now appreciate that now this 252 collection can be easily handled compared to 4,000 accessions. In case of wheat, we have around 2,300 accessions derived from around 22,000 accessions, and these cores are being maintained by our scientists, and they are being further studied for different purposes. This is the work on characterization in case of linseed, and we have strong funding from Department of Biotechnology, Government of India for this work. And these are the germplasm. This slide is on germplasm evaluation for different biotic and abiotic stresses, as well as the quality parameters. And during 1 year, we could evaluate our material for these traits across crops. Like in wheat, more than 3,000 accessions were studied. Barley, linseed, others, and other field crops. So evaluation of barley germplasm for major biotic stresses. I will not go into this detail, but just this slide I would like to share with you from this slide that as I mentioned, suppose you have some gene which is implicated in tolerance or resistance to particular disease, and it is a known gene, then you can profile your entire set of collection, and for the presence of that gene in under lab condition, and the presence of this gene in your existing germplasm can lead to the germplasm which are having which are expected to have drought tolerance, and then that particular set of material can be validated under actual field conditions. So these are the germplasm field days and awareness programs which we regularly conduct at our farms, and for almost all major popular crops, and a total of 6,900 accessions of wheat, barley, brassica, linseed, sesame, pulses, brinjal, and okra have been supplied to around 80 indenters from all over India during the previous year. This has stopped moving actually. Oh yes. This is the same slide, yes. And as I mentioned, our field gene bank facility was inaugurated last year by Honorable Director General ICR. He was accompanied by Deputy Director General Crop Science and Deputy Director General Natural Resource Management. And we had this program at our Isapur farm. The main activity which has been inaugurated there is related to the perennial species of this horticultural and forestry plants. And we received some of the national and international dignitaries to see our field experiments in Delhi. So during previous year, these are the varieties developed, especially in the case of potential crops in buckwheat, We have this Himfafra, Kunova entry, Himshakti, and winged bean, Chardari wall, Faba bean, and HFP2. So these have been released by the CBRC, and our scientists have and developed these varieties for potential crops. These are the genetic stocks registered in wheat and lentil. Similarly, in other crops also, we have registered and we are registering unique germplasm. Yes, so this work is performed through different institute as well as externally funded projects, and we have 12 institute funded projects in almost all feed and horticultural crops. Like we have project on characterization, evaluation, and documentation of feed, barley, and triticale. Then project on similar project on maize, pulses, oil seeds, vegetable fruits, medicinal and aromatic plants, abiotic stresses, wild species and pre-breeding, then potential crops, and now we have also project on agroforestry species. These are the externally funded projects which are running in our division, and a total of 10 externally funded projects funded by different national and externally funded agencies are in operation in division in different crops. So this just to sum up what we could do during the previous 10 years. So we have characterized and evaluated more than 1 lakh germplasm accessions for different agromorphological traits across different crops. A total of 11 core sets have been developed across crops. 76 germplasm are registered, and more than 50,000 germplasm have been supplied to different nearest partners as per their demands. 27 germplasm field days have been conducted, and 10 externally funded projects are in operation. So with all the trainees, I would like to share how we can try to facilitate you, how we can help you from NBPGR. So these are the areas where we can collaborate together, and we can share and exchange germplasm accessions because we are the government agency, and so there is not any inhibition in that context. Joint multi-location evaluation of identified material. We are already conducting this activity with our many nearest partners involving ICR institutes and the state agriculture universities, but this can be further, and this should be further intensified for joint work. Utilization of core sets, the reference sets, state-specific sets. We have already these core sets developed, and these can be shared for work. Collaboration for quality evaluation of germplasm. This facility we are having, and as per the specific request, we can also help them in this biochemical analysis. Then sharing of native germplasm for registration and documentation with PPFRA. We can also facilitate and guide the people on these aspects. In conclusion, this is my last slide. And characterization and evaluation have to be done in network mode, and utilization of improved techniques in characterization and evaluation should be followed, especially in view of the information science or digital science, information technology. Use of high throughput technologies for genotyping and phenotyping should be employed, and development of core and mini cores is very important so as to handle the entire set of germplasm. Characterization of crop wild relatives and their use in pre-breeding is also important, and detailed evaluation at hotspots for biotic and abiotic stresses. Whenever you find, come across and identify any germplasm, it should be tested under hotspot condition. I mean to say under high severity condition for that particular biotic or abatic stress. And then sharing of unique trait specific germplasm with the breeders should be done, and that we are doing already. So with this, I complete my presentation. Thank you very much.
Thank you very much, sir, for giving detailed account of revision of evaluation and national evaluation program of ICR and BPCR. So friends, as you know, evaluation holds highest priority among germplasm management or germplasm science, because until or unless we will not evaluate or identify the material, then it is of no use. So our motive is not just conservation in gene bank. Our motive is to conserve through use also. And sir, can we take some questions?
Yes, sure.
I am seeing in the chat box. First question is from Dr. Amar Khan, that is for annual crops, it is easy to characterize or evaluate the accessions, but how to proceed in case of perennial? So just, can I, sir, answer it?
Yes.
Because we have Dr. Amar Kant, we have also detailed lecture for, of course, what you said, indeed it is true that we have difficulty for characterization and evaluation in perennial crops. So we have also planned a specific lecture on tree crops. If you have seen in the schedule, that will be taken by Dr. Mahapatra. He'll be taking these things in detail. And second would be what is core and mini core from Dr. Vikas Pawar, and whether it is available for ICR NBP, for per millet in ICR NBP.
Yes, I'm able to read the questions now.
Please, sir, please.
Yes.
Please.
So the question is, what is core and mini core collection, and it is available with the ICR NVPGR for pearl millet. So far, we have not core developed in pearl millet, but this work can be done in that pearl millet that institute. So far, we don't have this pearl millet core. Yes. The next question is, what is difference between base collection and active collection? So, Dr. Kuldeep, would
Yes,
you like to?
Base collection is just, base collection, what we have kept in our gene bank, that is for long-term storage, that we are conserving at -18 degrees centigrade. And the active collection, which we are conserving at our medium-term storage, that is 4 degree. And active collection is, you can say that is working collection, what we are using regularly after, harvest, and then again we are resowing. That we are calling it active collection. So whatever indent we are receiving from indenters across the country, we are trying to, trying to supply from active collection, what we have in our medium-term storage.
So next question, what is the number of plants of perennial nature required for conservation of accessions? So
actually we have also a separate lecture for conservation, conservation for orthodox, recalcitrant, gene bank, if it's the schedule. But just to answer this question, it can be like we have very set standard for conservation of self-pollinated species, cross-pollinated species, seeds, perennial nature. Like for self-pollinated seeds, self-pollinated crops, we need to conserve around 2,000 seeds. And for cross-pollinated or often cross-pollinated, we have to conserve 4,000 seeds per accession. And for perennial nature, we have different national active germplasm sites in different ECRIP centers or institutes, where we are conserving the propagated material, like permanent establishing the field gene bank. There are many things, or we are taking the help from the cryobiology or tissue culture experts. Like this we are doing.
Yes, I'll take last question. Very nice. This one. What is most important trait of germplasm for evaluation and for biotic and abiotic stress? Yes. So this question is quite broad, but I would like to share with you that if we talk about any biotic stress, suppose biotic means disease, pest, but a disease. So, for example, then severity and this incidence of that particular disease, these are the important scientific traits which should be recorded for biotic stresses. As far as abiotic stress is concerned, there are different parameters to judge the abiotic stress tolerance, but depending upon your facility, you can employ different, maybe under controlled conditions, you can screen for abiotic stresses by employing, the stress under controlled condition. But under field condition, it is done normal. The abiotic stress tolerance is judged based upon their evaluation, not only under the stressed environment, but under non-stressed environment also. So based upon the magnitude of reduction under stress compared to normal condition, this stress susceptibility index is calculated. So accession which is having less this stress susceptibility index is considered more tolerant. So these are scientifically the better mechanisms to judge the stress tolerance. There was one question on yes, from core collection, how to go for mini core collection. As we mentioned, core collection is around 10% of the entire set of germplasm collection, derived through a set of methodology. But you further narrow down, suppose you have initially 1000 accessions, and based upon that you develop a core collection of 100 accessions. Then from 100 you further narrow down to say 8% or so. So it comes around 8 to 10. But as I mentioned, 1000 is quite less. So if we have like 5000, 10% of 5000 is 500, that is your core. Then 10% of 500 is 50. So your 50 will be your mini core. So I think that satisfies your question.
That is okay. And for just information of the participants, we have a specific lecture on germplasm utilization, and especially for the evaluation perspective, 2 more detailed lecture will be on germplasm utilization. That will be taken by the expert Dr. Jyoti Kumari, and another would be on descriptor development or guidelines. That will be taken by Dr. Ashok Kumar, who was former director and head of NBPGR Division of Germplasm Evaluation. So your query can be answered in those sessions respectively. So thank you very much, sir, for your detailed, thoughts, and you have also given a kind of, opportunities where we can collaborate, or all the participants in respective crops, they have opportunity to collaborate with NBPGR also on many aspects. And certainly we will be happy to, answer. We will be happy to collaborate, support, connect for the utilization of germplasm or sets. All information is available on the NBPGR websites, in which crops we have developed core set, mini core, or some publication you can get through. So thank you, sir. Thank you once again.
Thank you very much. All the best to all the trainees.
Thank you, sir.
Thank you.
And just for the information to participants, we will get back once our, another resource person, Dr. Vandana Madam, will join. We will get back to, we'll restart the session. If you have any other query, I will be happy to answer regarding. Acha, Vandana ma'am is joining. Okay. So friends, our next speaker, Dr. Vandana Tyagi, has joined. Ma'am, are you around? So friends, I would like to welcome Dr. Vandana Tyagi, madam. Madam.
Just a minute.
Okay, sure.
Yeah.
I would like to also take opportunity to introduce our speaker, Dr. Vandana Tyagi. Just I'm sharing one slide. Is it visible to everyone?
Yes, sir.
Okay, thank you. Dr. Vandana Tyagi is currently principal scientist and officer in charge of Germplasm Exchange and Policy Unit, ICR NBPGR, New Delhi. She has 25 years of experience in plant genetic resources exchange and PGR policy. She also facilitated the introduction of trade-specific germplasms for agri-horticulture crops. More than 1 lakh accessions from different agroclimatic zones were acquired in cereals, millets, vegetable crops, and medicinal plants. She also identified and introduced trade-specific germplasm for utilization in crop improvement programs of Indian plant breeders or researchers. She has organized various HRD, human resource development programs in germplasm exchange issues, regulations, and policies. She also acted as co-PI in one of the World Bank funded project on capacity building project for implementation of Cartagena Protocol on biosafety. She was also co-PI in FAO funded project on establishment of information sharing mechanism for monitoring the implementation of global plan of action. She is also member of the ITMC. She was also visiting faculty at Cornell University, United States of America from 2007 and 8 under Fulbright Hubert Humphrey Fellowships. She also published research papers that is amounted to 66, and besides this, she has also published other publications like 85. And friends, I would like to assure you all that I'm damn sure that this lecture will be your all cup of tea, because many times you need germplasm for your research and your breeding program. So this is very important lecture for all of you. So madam, like this, with this, I would like to welcome you.
Okay, thank you, Dr. Kuldeep, and thank you for your kind words and introduction. And am I audible to all?
Yes, ma'am. Yes, yes.
Yeah,
Please share
so
the slide.
I'll start my share my screen now.
Sure, sure. Put in slideshow, ma'am.
Yeah, it's okay.
No, no, it's not okay. It's not in slideshow. Just unshare and open your PPT.
Yeah,
then-
yeah, I have.
Now it's okay, ma'am.
Yeah, it's okay.
And
Okay.
yes, yes, yes.
So the topic given to me is introduction and exchange of plant genetic resources, and I'm working in this germplasm exchange unit since I joined ICAR. So I'll just I'll just start with a brief introduction about the crops which were introduced in dim past, since times immemorial. And if you say today, whatever our plant wealth is, it is result of plant introduction for several centuries. And if we see who introduced plants in the dim past, then all these have contributed towards introduction. Travelers, traders, explorers, and pilgrims. Now, they were doing it, and it was uncoordinated plant introductions since 19, I suppose 50s or 60s. But if we do have uncoordinated plant introduction, then there are some risk involved, and you lose some opportunities also. So to alleviate these risk and lost opportunities, plant introduction services were established in many countries. NBPGR is a plant introduction service. So, but some features which these plant introduction services have in common, I'll just elaborate on some points that whenever you hear of a plant introduction service, then these are the features which are common to those introduction services, like you have in USDA, USA, that they generally form part of the Department of Agriculture. They are a major research organization. Why? Because they need to draw extensive resources carrying out their work. They have direct responsibility for plant quarantine, like we have in NBPGR, or they work in close collaboration with the quarantine authority, like the one you have in USDA Maryland office, which is APHIS, Animal and Plant Health Inspection Scheme. They should have at their disposal good testing facilities in the major agroclimatic zones of the country. Like we have at NBPGR, our 10 regional stations in different agroclimatic zones. So whatever we are introducing or importing are being tested at the similar type of zone. They need to maintain records of introduced plants so that we avoid unnecessary duplication. And we have that also in place at our bureau. And by maintaining these extensive collection and by establishing close collaboration with their counterparts, the service is able to provide plant material needed by researchers. And they also provide a contact point for the international collaboration, either through FAO or other agencies. if you see how this plant introduction service was established, if you go to the history, then in 1935 it was at conceptual level, and in 1941 the need was reiterated, and then it came up with the plant introduction scheme in 1946 at Indian Agricultural Research Institute, USA. It was upgraded in the next 5-year plan as Plant Introduction and Exploration Organization. And finally, a division of plant introduction was there in IARI. And in 1976, it was upgraded to an independent institute, National Bureau of Plant Introduction, which was renamed in 1977 as National Bureau of Plant Genetic Resources. So ICR NBPGR is the only institute authorized for introduction and maintenance of germplasm of agricultural and horticultural crops. Anyone can import, individual scientists, universities, other research organizations, they can also introduce. But all the introduction in India are routed through NBPGR, New Delhi. And how to define an introduction? What is a plant introduction? Plant introduction is when you take a genotype or a group of genotype into a new environment where it has not been grown before. That is introduction. Generally, we refer to it whatever we import from outside, abroad. But if you introduce anything from one state to another state within India, then also it is an introduction. Because introduction means you are growing something into an area where they were not grown before. And they may include new varieties of crop, wild relatives, a totally new crop species. And based on the utilization, you will define it as primary or secondary. You must have all read through in your syllabus. When you introduce a variety, it is well suited to the new environment, and you release it for commercial cultivation without altering any of its genotypes. That is a primary introduction, which is less common in the country which have well-organized crop improvement programs. However, we have some examples. Secondary is when you introduce something, when you introduce a variety or a genotype and you select it to isolate a superior variety, or you hybridize it with another local variety, or you transfer one or more characters to the variety to the local ones, then it becomes a secondary introduction. And examples are many varieties like Pusa Ruby, Pusa early dwarf tomato, Sonalika, Khadchia, they are all secondary introductions. Now, what is the ultimate aim of introduction? Why you want to introduce or import? Ultimate aim is you want to improve your plant wealth. So either you want a new crop or a new variety. You want to have a diverse gene pool. You want to hybridize them for crop improvement programs, use them in research programs. You introduce CMS restorers to make hybrids, or you want to save them from diseases and pests. That is the ultimate aim. So we at NBPGR are authorized to introduce or import, and exchange of PGR activities at Bureau involves 3 major activities. One is import, then export, and also meeting the needs of the researchers within the country. We call it as national supply or domestic supply. I'll just give you a brief of these procedures, and my colleagues, Dr. S.K. Yadav and Dr. Pragya, will be taking practical in the afternoon. They will be explaining everything in detail. So if you see import, we are governed by Plant Quarantine, which is Regulation of Import into India Order 2003, but we refer to it as generally PQ Order 2003. And it is available online at Plant Quarantine site. NBPGR is doing for research purposes and in small quantities. While if you wish to import in bulk quantities and for commercial aspects, then the agency is Directorate of Plant Protection, Quarantine and Storage. The office is in Faridabad. And for research purposes, NBPGR is the nodal institution. As per the order, clause number 6(2), and Director NBPGR is authorized to issue import permit. And whenever you want to introduce anything into the country or you want to import anything into the country, then there are two mandatory requirements which are to be fulfilled for every material which is coming into India. One is you need to have import permit before importing any plant material, and you also need to have phytosanitary certificate from the country of origin. Like if you are importing anything from USA, then import permit from NBPGR and phytosanitary certificate from USA. So the procedure is detailed. Our colleagues will also take, but for just briefing that about it, you need to apply in a proper application form, which is called PQ08. PQ is plant quarantine. So they have different forms in their order. So PQ08, which you need to fill, and then you have to apply to Director NBPGR. You have to send this application to a Director NBPGR. And if you are a private seed company, if anyone is from private seed company, then they have to submit a recognition certificate of Department of Scientific and Industrial Research, that their research and development is recognized by them. And we are now doing it online. So if you go to NBPGR website under germplasm import, you will find this apply for import permit. You can go there. There's a small registration form which you need to fulfill. You have to first sign up, and then once your account gets activated, you have to log in. Detailed, my colleague will be explaining in the afternoon. And when you log in and you get the same PQ08 of performer online, you can fill it and submit. And then this import permit in triplicate, 3 copies will be issued to you. This one is a duplicate copy, one is original, and one is duplicate, and one we retain as an office copy. Now there are different schedules under this plant quarantine order. There are schedule 4, 5, and 6. Schedule 4 list plants where import is prohibited, and justifications are also given. Like some crop is a restricted import from certain country because of risk of certain diseases. And schedule 5 list plants which you can import only with the recommendation of authorized institution, which are given in that order. Schedule 6 list plant material which you can import with additional declarations and special conditions. This additional declaration means they have to be certified that they are free from certain diseases and pests. And the fee structure is also there for import fee, which you can pay online. And this is also payment gateway is available at NBPGR website. You can pay there the fee. You can select what type of fee you are going to pay. Quantity is also defined and approved for these import. Now this was just import permit procedure, but if you wish to import any transgenic, then there is one additional step before applying in PQ08 form, that you require approval of Review Committee of Genetic Manipulation, which is an authorized agency under the Government of India, functions under Department of Biotechnology, and handles the import of transgenic material. Whenever any institute who is working on a transgenic, at that institute, you will find they will be having an Institute Biosafety Committee. So when you apply to RCGM, based on your application submitted by your Institute Biosafety Committee, will consider only those applications. So you cannot directly approach RCGM. You have in your institute an Institute Biosafety Committee. You apply to them. The Institute Biosafety Committee will send your proposal to the Review Committee on Genetic Manipulation, or RCGM. This RCGM have members from Indian Council of ICAR, ICAMR, Indian Council of Medical Research, Council of Scientific and Industrial Research, CSIR, Department of Biotechnology, and other expert in the individual capacity. And generally they will hold meetings once in 2 months or every month, depends on the number of applications they receive. Sorry. So what you have to do is, when you need to have a permit letter from RCGM and apply to Director ICR and BPGR in the prescribed application form, which I earlier told you, that is PQ08. When RCGM gives you permission, there is a para there in para 6. Para 6 says that some voucher specimen will be retained by NBPGR, and you have to submit some certificate that the transgenic which you are importing does not carry any embryogenesis deactivator gene. So you have to fulfill and furnish those information which are as per para 6 of DBT, and there are some instructions for use also. So you have to certify and give that certificate to NBPGR or to RCGM, and then submit the application to Director NBPGR. So the same procedure is there. When you submit the application for transgenic to NBPGR in PQ08 form, you have to enclose the permission of RCGM. You have to pay the fee as prescribed for the transgenic material, and then import permit will be issued in PQ in triplicate copies and sent it to you, 2 copies. Now in DBT in January 2020, it simplified some procedures for import of transgenics. So if you are importing any model organism, such as plants like Arabidopsis or other model organism, which are routinely used in laboratories, then we issue import permit based on the certification of institution by safety committee only. So your institute by safety committee will give certification, and then you can apply with that institute by safety committee certification. Don't need to contact RCGM for such model organisms. And your institute by safety committee is required to certify that the model organism carry routine and standard experimental insertions and do not carry any foreign gene insertions from non-model organisms. Once you have that import permit in place and phytosanitary certificate, then it is required that when you send the import permit to the concerned agency, consignment must be addressed to Director NBPGR only, as the port of entry is New Delhi only. Along with that, you required to fax the details of the shipments, AIV bill number, date of shipment, custom value, invoice copy, etc., which are required. If you get material directly in your name, it will not be released by custom people, and you will face a lot of difficulties, and most of the time the material will be incinerated. So it's always make sure that the material should be addressed to Director NBPGR only. In any chance, however, if by courier or etc., you get the material directly in your name, the first thing is you should send all the papers and the materials to NBPGR for quarantine examination. Now I just told you initially that all the information is documented with the NBPGR. So if you go to this website, germplasm exchange and quarantine information system, you will find this. This is free for anyone can access this website. If you find on the right, you will find this EC search. And if you click on that, you will get a form like this. So if you have any information with you, like you retrieved any material in this date, if you know the IQ number, you know the IP number, you know anything about these fields, you can just put it there. Variety you have introduced, name, you can name the variety. You click here, select all, generate report, you will get all the information about that material which was introduced. And this database includes all the material which were introduced or imported since 1941. Now, I'll just come to the export part. Okay. Now, Dr. Pratibha, you must have heard the lecture of Dr. Pratibha. Briefly, she explained you about all the conventions and the treaties and the agreements which are in place regarding access of germplasm from India. However, I will give you in detail something and some little bit explanation of all these things. She already told you that before 1993, if you say before CBD came, it was free. Access was free because the plant genetic resources or the biological resources were considered as heritage of mankind. Now, then came CBD in 1993, and it established the sovereign right of nations over their biological resources. It also said that you have every country which is a contracting party to the Biological Diversity Act have to conserve, have to fulfill all the objectives of the CBD, and they have to either they should adopt as such or they have to develop their acts in conformity with the CBD. And every access should be regulated with prior informed consent on a mutually agreed terms. So India came up with this Biological Diversity Act in 2002. And access is now regulated by Biological Diversity Act. Side by side, under International Undertaking on Plant Genetic Resources of FAO, the concern was if you regulate all the biological resources or even plant genetic resources, then what will happen to the plant genetic resources which are very crucial for food and agriculture. She explained to you in detail what are PGRFA. So for those PGRFA, international treaty was signed. It is known as International Treaty on Plant Genetic Resources of Food and Agriculture, and generally we refer to as a treaty. So they established a mechanism of MLS. And some crops were identified based on consensus. 64 major crops are part of this MLS, which are very crucial for food and nutrition security. For those crops, the countries who are contracting parties are asked to provide facilitated access, and every access should be with the signing of standard material transfer agreement. Now, first I will just go into the Biological Diversity Act. I'll just give you all those important sections which are of use to you, which are of concern to you. Biological Diversity Act came in 2002. It was made in compliance to CBD because it says you have sovereign rights over your biological resources, and India was signatory to the convention. So it promulgated the Biological Diversity Bill, introduced it to the Parliament, received the assent of the President, and then it came as a statute book as Biological Diversity Act. The rules came in the Act came in 2003, sections came in 2040, rules. Why it was enacted? Because it was enacted in a consequence of the fact that we are rich in biological diversity, traditional knowledge. India is party to United Nations CBD. CBD affirms the sovereign rights of the states. So it was very important to provide for conservation, sustainable utilization, and equitable share of benefits, which are the prime objectives of the CBD. And if you look at the Biological Diversity Act, it is a 3-tier system. At the center, you have National Biodiversity Authority. The office is located in Chennai. And it is in the Act only that states that the office should be outside Delhi. So it was based in Chennai. Then every state has a state biodiversity board, and you have biodiversity management committees at local and panchayat level. Any access by any foreigner, which I will say who is a foreigner, it is defined under the Biological Diversity Act. So if any foreigner access any biological resource from India, it is regulated by National Biodiversity Authority. Any Indian for commercial use want to access biological diversity, it is being taken care by State Biodiversity Board. And for commercial purposes, if any access is for commercial purpose, either by foreigner or for an Indian, then it is taken in consultation with biodiversity management committees. 5-6 important sections I need to tell you here. One is section 3. Section 3 is the most important section of this Biological Diversity Act, where it defines who is a non-Indian. And it says any non-Indian or a non-Indian entity, if they want to access any germ plants from India, and it doesn't say India, it says occurring in India. So any non-Indian entity, if they want to access any biological resource which is occurring in India, prior approval of National Biodiversity Authority is mandatory, is must. And then they have defined who is a non-Indian. It says a person who is not a citizen of India, is a citizen of India but is a non-resident, a body, corporate, association or organization which is not incorporated or registered in India, or if incorporated or registered in India, has any non-Indian participation. And it says any. They have not defined what is any. So even if a single rupee participation of a non-Indian company is there, it becomes a non-Indian. All multinational companies which are registered in India, all private seed companies, they are non-Indian in the eyes of Biological Diversity Act. And they cannot access any germ plants without the approval of, prior approval of National Biodiversity Authority. Section 4 says, Section 3 is saying non-Indians cannot access any biological resources. Section 4 says you cannot even transfer your research results. So a non-Indian entity, if they want to have your research results, they are required to take prior approval of National Biodiversity Authority. Transfer of results of any research which is relating to any biological resource which is occurring in or obtained from India to any person who is covered under Section 3 means who is a non-Indian for monetary consideration or otherwise. Only exemption is you can publish research papers, you can disseminate knowledge in any seminar or workshop, and if the publication is as per the guidelines which is issued by central government. There are guidelines which are available at an NBA website. However, there is an exemption under Section 5. That exemption is for collaborative research projects. So if you have a collaborative research project that involves transfer or exchange of biological resources, it should be between the governments, both. Like if you have a collaborative research with USA, then it should be by US government, approved by the US government. Then those Section 3 and Section 4 will not apply. But those collaborative research projects must conform to the policy guidelines which are issued by the central government. In this behalf, these guidelines are issued in 8th November 2006 and are available at NB website and approved by the central government or the state government, whatever. So you have to, that is the only exemption provided under Section 5. Then Section 6 says, if you are applying for intellectual property rights of any material which you have accessed or obtained from India, this is for Indians also. No person can apply for any IPR outside India or inside India for any invention which is based on research or information on a biological resource which you have acquired or obtained or from India. Then before making such application, you need the approval of National Biodiversity Authority. If you apply for a patent, permission may be obtained before the acceptance of the patent, after the acceptance of the patent, but before the sealing of the patent. If you by any chance you have applied, you have not sought approval of National Biodiversity Authority, then what you need is before sealing of the patent, you should take the permission. Why these all things? Just you want to have equitable share of benefits. The benefit should flow to the person from where you have acquired the biological resource. And then section 6 says, if you are an Indian, you want to do a research, you can have material from India. But if you are an Indian and you want to use biological resource for commercial utilization, then you have to intimate first to State Biodiversity Authority. This section does not apply to local people and communities of the area, Veds and Hakims, who have been practicing indigenous medicine since ages. But this says local people and communities of the area. It doesn't mean that a Ved practicing in Uttarakhand can access biological resources from Gujarat. So what are the exemptions under the BDA? BDA, it says you have to take approval of National Biodiversity Authority. Certain exemptions are there. So what is excluded? Under section 7, local people and communities of the area, growers and cultivators of biodiversity, Ved and Hakims are exempted. Under section 5, collaborative research projects which are conform to the policy guidelines are exempted. Under section 40, there is a section 40 where you can provide some exemptions. Section 40, normally traded commodities, which is the material used as a commodity, are exempted, like we export lots of basmati rice, spices, many things. When this treaty came up, we had Biological Diversity Act in place. It says you have to regulate access, but the treaty says you have to provide for facilitated access. Then to bring in harmony with these two acts, we wrote, NBPGR had many conversations with National Biodiversity Authority, and they issued a notification in 2014 where Annex 1 crops or MLS crops of treaty were exempted from the purview of Biological Diversity Act. So if you now have any request under international treaty, you can facilitate the supply. And this mainly we are doing it for ECRISAT export. ECRISAT is an international center, CG center, which is located in India. They have to comply to the request of many countries, supply material, but they are housed in India and they have to be regulated by Biological Diversity Act. So this exemption was given by National Biodiversity Authority. Indian citizens, entities can do research, no problem, they are exempted. Publication of research papers, presentation in seminars, workshop is excluded. Conventional breeding or traditional practices in horticulture, agriculture, poultry, dairy farming, animal husbandry, for beekeeping in India are exempted. This was issued in the notification of December 2014. So based on these acts and conventions, we have divided our germplasm export into 4 categories. One is export of Annex 1 crops which are under treaty, and FAO designated accessions of CG centers located in India. The FAO designated accessions means accessions which were collected prior to CBD, collections before 1993. They were held in various gene banks, international agriculture research centers. So for these accessions, Biological Diversity Act or CBD does not play any role. So they are FAO designated accessions. No one can have IPR on them, and they have to be provided facilitated access. Category 2 becomes germplasm, which are under collaborative research projects, because sections 5 provides for exemption for collaborative research projects. Then in a notification which when Nagoya Protocol came and for ABS guidelines, under this in 2014, another exemption came from NBA. Any Indian researcher or a government institution who wants to carry or send germplasm for non-commercial research or research for emergency purpose other than collaborative research becomes a category 3. Actually, this came up after several discussions, because if you see Biological Diversity Act says you have to give permission to take permission from NBA, and many students were going on many fellowships outside India. They would need to take some germ plant, and but they were not able to do so. So this clause came up, and for this they have established a timeline. Within 45 days, they give you approval. Then anything which is not fitting anywhere here, you have to apply to National Biodiversity Authority. As of their timeline, they give approval within 6 months, but it does take time to get the approval from National Biodiversity Authority. Now I'll just give you a briefly about how you export under these different things. Now, this is under treaty. Implementation of international treaty is less with Department of Agriculture Cooperation and Farmers Welfare. So, any request under MLS of the treaty, the nodal agency is DAC. The applicant is required to approach DAC, which is the national focal point. NBPGR gives recommendation for such export. Once they give permission, NBA is informed accordingly. If you have a collaborative research project as per the guidelines, then all that export application are addressed to Director NBPGR. We seek recommendations of the concerned crop director, project coordinator. Director NBPGR also gave recommendations, and SMD or DARE gives approval for that. For Biological Diversity Act implementation or CBD implementation, Ministry of Environment and Forest is the nodal agency. Detailed procedure, my colleagues will be giving you. Then the third one, where the students go for fellowships and any programs. This processing application, you have to apply in Form B. You have to seek approval of National Biodiversity Authority. You need to apply in Form B. Along with that, you need to submit an undertaking. They have given you that within 45 days, they will give you approval. But generally, they give in 15 to 20 days. For all this, which is not fitting into category 1, 2, 3, you have to apply to National Biodiversity Authority. There is a Form 1. You have to fill Form 1. There's a fee, and it is available online. You can go online and fill in that form. They say approval within 6 months, but generally it takes more than 6 months. Now, the third part is when you want to indent any germ plant from NBPGR, National Gene Bank, active collections or base collections, or through any active germ plant site, then you have to send your request in the requisition performer, which is called JEX01. It is available at NBPGR website. You have to fill in that, sign the material transfer agreement. This is that JEX01 form. This is MTA, material transfer agreement, and submit it to Director NBPGR. We make sure that you get the desired material and in very short time. So all those private seed companies which do not fall under Section 3(2) of Biological Diversity Act means they are wholly Indian. Section 3(2) defines who is a non-Indian. So if you do not fall under Section 3(2), it means you are a wholly Indian company. So for those companies also, we are providing the material germ plant. Only thing is we require from them an undertaking on stamp paper of ₹50, certifying that company does not fall under Section 3(2) of PDA. And they are also required to submit the DSIR certification that their research and development is certified by DSIR. That's all for. And now I would like to have questions, if you please.
Hello.
Hello,
Good morning, ma'am.
good morning.
This is Kirti Rani from Regional Station Jodhpur. Ma'am, while I was posted at DGR, Directorate of Groundnut Research at Junagadh, we got one request to export the groundnut germplasm from the ICRISAT. So that you have mentioned now that FO designated material can be exported, but what about the breeding materials that have been developed after the FO after the CBD, but using the FO designated materials? And what about the breeding materials developed using material other than FAO designated material, but not Indian origin? These was the few queries that that time I was not clear of. So please explain.
Yeah, when you, if you see in, any application which ICRISAT submits to NBPGR for export, they categorize their material into 4 different categories, whether that is FAO designated or developed using FAO designated material of Indian origin or non-Indian origin or exotic. Only the ones, everything can be exported. We have a system in place. We check the designation of that FAO status. So you put that accession number in the system, it will say it is FAO designated. Okay. Then if it is FAO designated non-Indian, FAO designated developing using FAO, not FAO designated, but developed using FAO designated, exotic, we just define that it is FAO designated or non-FAO designated, but developed using FAO designated, or it is exotic, Indian, but developed using non-Indian developed FAO designated, or Indian developed using FAO designated. All those categories are there. Okay, but everything for that, it is processed, recommended for export.
So all the breeding materials can be exported.
Yes. All breeding materials and FAO designated accessions which have FAO designated status either as a parent in their pedigree, either as a parent or itself it is a FAO designated accession, they can be exported. We send recommendations through directors concerned. Like if you get a groundnut request for a groundnut export, we send those all details to Directorate Groundnut Research for their recommendation. So if they will recommend that, okay, you can send this, we send that recommendations to DAC. However, if Directorate Groundnut Research has something about that Indian ones which are developed using FAO designated, they can restrict that export. Otherwise, the breeding material which does not use any Indian origin material, which is not FAO designated, can be exported.
Okay.
Which is not FAO designated are restricted for import. Otherwise, everything can be exported which have FAO designation in its chain.
Okay. Thank you.
Okay.
Any other questions?
Hello, ma'am.
Please.
Hello.
Just
Yes.
Hello.
introduce also yourself before asking.
Hello.
Yes, yes.
You are audible.
हाँ,
You are audible.
Myself Dr. C.S. Pandey from JNKV जबलपुर. मेरा क्वेश्चन मैम ये है कि जैसे ये जो एक्सपोर्ट इंपोर्ट जब हम रिसर्च के लिए करते हैं और अदर देन रिसर्च जो हम एक्सपोर्ट इंपोर्ट करते हैं तो दोनों में क्या डिफरेंस है?
किसमें
प्रोसीजर में क्या डिफरेंस है? जैसे कोई रिसर्च करने के लिए हम कोई चीज इंपोर्ट कर रहे हैं या कोई प्लांट मटेरियल एक्सचेंज कर रहे हैं, तो ये तो आपने डिटेल में बताया, लेकिन अदर देन रिसर्च पर्पज अगर कोई एक्सपोर्ट इंपोर्ट हम कर रहे हैं तो उसके लिए क्या मतलब बेसिक गाइडलाइंस हैं? या क्या
अदर पर्पस के, अदर पर्पस,
जी हम ये जानना चाहते हैं गवर्न कहाँ से होता है? ये अपने एनबीपीजीआर से तो रिसर्च वाला गवर्न होता है टोटल, लेकिन अदर देन रिसर्च मटेरियल ये कौन सी बॉडी गवर्न करती है, कैसे होता है?
वो मैंने आपको शुरू में स्लाइड में बताया था, जब हम बल्क क्वांटिटी कमर्शियल के लिए करते हैं तो डायरेक्टरेट प्लांट प्रोटेक्शन क्वारंटाइन एंड स्टोरेज के थ्रू होता है, उसमें डीएसी के थ्रू एग्जिम कमिटी होती है, एक्सपोर्ट इंपोर्ट कमिटी।
राइट।
प्रपोजल जो है, जब आप अपना प्रपोजल बनाएंगे तो
जी
वो प्रपोजल आपका एक्सपोर्ट इंपोर्ट कमेटी के पास जाएगा,
ओके ओके
जो कि डीएससी के अंदर है।
राइट राइट
डायरेक्टरेट प्लांट प्रोटेक्शन क्वारंटाइन एंड स्टोरेज उसको देखता है।
जी मैम
तो जब आपकी एप्लीकेशन वो होगी, उसमें आप जब सबमिट करेंगे तो वो एक्सपोर्ट इंपोर्ट कमेटी में डिस्कस होगी।
ओके मैम
वो
ओके
हर मंथ के एंड में एक्सपोर्ट इंपोर्ट परमिट की कमेटी की मीटिंग होती है, जिसमें एनबीपीजीआर का मेंबर भी जनरली एक्सचेंज से या क्वारंटाइन से एक पर्सन उसका मेंबर होता है।
ओके मैम
फिर
ओके
वो आपको ना, जैसे आप अगर फर्स्ट टाइम मंगा रहे हैं वो चीज, तो आपको केवल ट्रायल पर्पस के लिए आपको परमिशन मिलेगी।
ओके मैम
आपके
ओके
कोई सर्टिफिकेशन मांगे जाएंगे कि आपके पास क्वारंटाइन फैसिलिटी है, नहीं है, तो ट्रायल पर्पसेस के लिए आप मंगाएंगे। उसके बाद आप उसको बल्क क्वांटिटी में मंगा सकते हैं। और जो ऑलरेडी उनकी शेड्यूल लिस्ट में है, जो आ रही हैं ऑलरेडी, उनको आप बल्क में भी मंगा सकते हैं।
ओके ओके
लेकिन
मैम
आपके
अपीडा को
डिस्पोजल पे प्लांट क्वारंटाइन फैसिलिटी होनी चाहिए।
जी अपीडा का क्या रोल है इसमें मैम? यहाँ पे कुछ है इसमें एक्सपोर्ट
नहीं,
इंपोर्ट?
अपीडा का कुछ रिस्ट्रिक्टेड जो एक्सपोर्ट है या कुछ है उसको देखने के लिए है, वो भी बल्क में ही और कमर्शियल वाले एस्पेक्ट को ही देखती है, रिसर्च के लिए नहीं। रिसर्च का पूरा एनबीपीजीआर के पास है।
हाँ यस मैम वो तो आपने क्लियर बताया जी
और बाकी नॉर्मली ट्रेडेड कमोडिटीज भेजते हैं,
ओके
वो अलग डिपार्टमेंट है, मिनिस्ट्री ऑफ फूड एंड कॉमर्स।
ओके मैम ओके ओके हाँ थैंक यू मैम।
Can we take from chat box?
या
They have Dr. Vikas Pawar asked that, and whether NBA have any office at regional level?
नहीं, शुरू में जब वो स्टैब्लिश हो रहा था तब उनका एक छोटा सा एक फैसिलिटेटिंग यूनिट था यहीं दिल्ली में एनएससी के पास। अब नहीं है, अब उनका खाली रीजनल ऑफिस की जरूरत नहीं है। एक्चुअली एवरी स्टेट हैज अ बायोडाइवर्सिटी बोर्ड, एंड at local and panchayat level you have biodiversity management committees which maintain records of all biological resource occurring in that area. So all state biodiversity boards are functional, union territories are governed by NBA, and every state biodiversity board had BMCs in place. There is no regional office. Centrally you have NBA, and then every state has a state biodiversity board.
One more question, like for exporting a research material to any other international trials, for example, they have written like advanced rice cultivars to Erie, Philippines, whether it comes under category 2 or 3.
If you have a collaborative research project in place, it comes under category 2. Otherwise, you have to seek approval of National Biodiversity Authority.
Okay, and another question by Dr. Padmaja from PGTSAU. How we can import non-shattering sesame, the sesame germplasm for machine harvesting or like this?
How we can import from? How I can, please pardon?
Sesame, it's for the general.
What question?
Sesame
Please
crop,
repeat
sesame.
once again.
How we can import non-shattering sesame suitable for machine harvesting? Or some varieties she is asking the procedure, I think.
The procedure is the same.
I think she is willing to know the basic work has to be done by her, all we can do based on her request like this.
I didn't understand the question actually.
Actually, what I think she is willing to know, let's say she has to introduce some trait specific varieties.
Okay.
So that work, who will identify that? If she will identify, then we can import, or we can identify ourselves.
She can import, she can identify, she can work on that.
She can request to us.
If she wants, she can request to us.
Okay.
We can go either way you can do it.
Okay. Any other question from participants? Because this is the most, I think, one of the most important lecture, because everybody is indenting germplasm from NBPGR. And one more thing, what I feel, feedback is also important thing, madam, I think.
Yes. My colleagues will be telling when they are taking practical examination, they will ask you to provide feedback information.
Okay.
Feedback information and even multiplied seed material, because whatever we are importing, we are sending to the indenters, because it comes in very small quantity, and we ask them to multiply the seed material and send for deposition in National Gene Bank, so that we next time they don't need to import, we can supply from Gene Bank.
Any other question from participant side? If no, then ma'am, thank you very much for a very informative lecture. I hope they all will be familiar with the procedures, and basic things or basic guidelines from import and export. So thank you once again, ma'am.
Thank you, Kuldeep. Thank you, organizers, and thank you, everyone. Thank you, participants.
And just for information of the participants, we will start from 12:15. And in that session, we will have introduction.
No, one question from chat box. What is the procedure
Okay,
for
please,
coconut
please.
variety import from Indonesia? The procedure does not is not specific to any country or any crop. Procedure is same for every import. Only thing is, if you are importing coconut, first you should have recommendation of CPCRI for import, because it's a central institute for that, and then submit the PQ08 form to Director NBPGR. Procedure is the same for any crop or any country. Only thing you have to check as per the schedule whether the import is prohibited or whether it is requires a recommendation of a central institute, or whether you require some certain justification for additional declaration, etc. That's all. Otherwise, the procedure is the same.
Madam, sir,
Yes.
Please.
sir, one doubt, myself, Dr. Ramesh. Madam,
Yes.
who is the chairman of the State Biodiversity Board, madam?
The chairman is for National Biodiversity Authority.
Madam.
State biodiversity boards will have
Which ministry comes under in state level? They are independent body, madam, this is?
state biodiversity boards are independent, autonomous.
Okay, madam.
And NBPGR officer in charges of regional station are the member, I think, madam. No?
State
Some of
board
the-
biodiversity boards.
State biodiversity board. Wherever the NBPGR regional station is there are the members from NBPGR and state biodiversity.
Exception, madam. Okay. Right, sir.
Okay. And thank you, madam. Thank you.
Okay, thank you, Kuldeep.
Unmute कीजिए.
Mute yourself.
Okay.
Unmute
We will
yourself.
have, sorry. We will have an introduction, and introduction, we have to be very brief, like name and, from where you belong, or what kind of nature of work you are doing there, from which institution belong, just to know each other, and we can seek the opportunities where we can collaborate with you all, and you also will be familiar with me. So we will start on 12:59.
Kuldeep, we may start later on if
कब बताइए मैम?
because afternoon visit is there ना, तो 1:30 तक, otherwise फिर
12
delay हो जाए.
12
12 कर
is
लेते
okay?
हैं, 12 to 1
12.
कर लेते हैं.
Okay, thank you.
Okay.
From last 2 days, we have gained much more knowledge about all these things. So I would like to thank the organizer also. Thank you.
Okay, thank you, Dr. Amit. And Dr. Amit M. Polara from Junagadh. Okay, Dr. Anishetty Sarita.
Good morning, sir.
Please,
Hello.
please, madam, please. Yes, you are audible.
Good morning, sir. I am working as a scientist, plant breeding at PJTSAU, Telangana, sir. Actually, currently I am working as a maize breeding, sir. From this Kharif 2022, I am working as a for evaluating the germplasm and trials in grain amaranth, sir, under ACR and potential crops, sir.
Okay.
Actually, I need the maize breeding material, sir, for breeding purpose at PJTSA, sir. Please give the procedure how to explore that material from NBPGR, sir.
And that will be taken in the lecture. But for your information, Dr. Jyoti Kumari is here. She is taking care of the maize germplasm, so you can discuss with her directly or indirectly. And
Yeah, you can discuss about your kind of requirement, and based on that you can make the indent.
But not here. You can discuss in detail
Okay.
with her.
Okay, sir. Okay, sir. Okay.
Thank you.
Thank you, sir. Thank you, madam.
Thank you, madam.
Okay.
And next is Dr. Anuj Kumar Singh
Okay.
from SDAU, Dantiwada. Dr. Anuj, are you around? If no, then Dr. Vilas Ramrao.
Sir, good afternoon. Am I audible?
Yeah, very much.
Okay. So myself, Dr. Vilas Awari. I'm working as assistant professor in agricultural botany at MPKV Rahuri. I'm associated with teaching as well as I am handling the one state level gene bank is established at MPKV Rahuri. So I'm looking after this activity, and at present now we are collecting the germplasm from the breeders of our university, and then we will collect from the universities of our Maharashtra agricultural universities, and then we will conserve it. So just it is the initiative movement that our university has started.
Okay.
And also this training will be helpful for me, and also I will request to all of you, please allow our team to visit at NBPGR, and at least 1 week training is required.
That you can give your suggestion during the feedback session, or you can give. We will identify some of the basic requirement from your side, what you are expecting from us, that you can directly tell in the last session when we are having feedback, or and anytime you can request to the director NBPGR for these kind of things. There is no problem at all. Always NBPGR is open for everyone.
Okay, sir. Thank you very much.
Thank you, Dr. Vilas Ramrao. And now Dr. Aisha Mohanty from OUAT. Dr. Aisha, yeah, please.
Good afternoon, sir.
Good morning, madam. Good morning.
Yeah, I'm Dr. Aisha Mohanty. I work as an assistant professor here at OUAT College of Agriculture, Chiplima. So I'm really very sorry for the
No problem. We understand. No problem. Please.
No, that's fine. You can carry on.
Yeah. I'm sorry. Okay. So I am actually sorry.
We can come to you after some time. No problem.
No, it's okay, sir. It's okay. I'm fine.
Please. Bataye.
So I have a baby actually, so I have to.
No issue. You can.
Thank you so much.
Please.
So I work basically in the field of soil science, and I'm more interested in research nowadays, and about microbiology and all. So I think this, this training will be much more beneficial to me. And another course was there in data analysis and all. So these things will help me to guide any students or something in future in this. So I just enrolled in this training for my own benefits and for my career growth.
Okay, madam. Just we are very much curious to know the expectation of yours from our side.
Yeah, so that means the time is there, so we can incorporate in between some correction measure we can take up during.
Also, because we all are working on some specific crops. So let's say somebody told that she's working on maize. So we have Dr. Jyoti Kumari with us, and she's dealing with the maize. You can get the opportunity to interact, connect, collaborate, and work all together. Whether it's a student program, or you can also find suitable person in NBPGR for particular research purpose. That's why this is the
regarding your expectation data analysis. So we have full day class on data analysis one day.
Yes.
So you can get your queries cleared that day.
Okay.
Yes, of course.
Thank you, Dr. Mohanty. Thank you. And
thank you so
now Dr. Bangarema Wadiar from College of Agriculture, Vijayapur. Dr. Wadiar.
Good afternoon. Good afternoon to all.
Please, madam, please.
Myself, Bangarama Wadiar. I'm working as assistant professor at College of Agriculture, Vijayapur, which comes under the University of Agriculture Sciences, Dharwad. I'm now in teaching, sir, actually, and parallelly I'm working on horse gram and moth bean. Okay, from this course, I want to learn the procedure and protocol for borrowing the TGRs. That is the thing, sir.
Okay, ma'am. Thank you. And just for information, we have developed the course set in horse gram and moth bean both.
So actually, I have borrowed some things from IPR Sub-Center, Regional Center, Dharwad, sir, last year. I have evaluated 125 germplasm lines of horse gram already.
Just for information, we have around 4,000 accession of horse gram and around 2,000 accession of moth bean. And we have also developed the course set that you can use for your breeding program and this thing.
Sure.
Okay, ma'am.
Thank you.
And Dr. Bhavna Sharma from IGKV, Raipur. Dr. Bhavna, are you around? No. And Dr. Chandana Behra, please. Yes, ma'am, you're audible.
Am I audible, sir?
Yeah.
First of all, very good morning to all of you. And I'm very happy to join this course because I did my PhD from IRI, and Jyoti madam, she also took some classes for us. I am the student of
I remember that. Yeah, by seeing your name, I could recall that. Okay, okay, good,
Yes,
good.
ma'am. And Dr. D.K. Yadav sir was my chairman during my PhD work. And at present, I'm working as an assistant professor in the Department of Genetics and Plant Breeding, College of Agriculture, Bhavani Patna, OUAT. So here till 6th year, I'm in teaching only, that too only UG course. So I think that I will just join this course, this training program, by which I can just have some idea regarding the plant genetic resources. And here lot many variations are there with respect to the crop, because where I have posted, that is Palahandi, one of the KVK region of our Odisha. So here we can find lot many variation with respect to the different crops. So here we can just through that particular training program, I can know about the germplasm collection, conservation, characterization. So this is my main objective to join the training program. Thank you all.
Sir, good morning to all. Can I introduce myself?
We are calling one by one, madam. Na? Hello. We have power cut if I am audible. Okay. Am I audible?
Yes,
sir,
you're audible.
Thank you. Here is power cut. That's why. Okay, no problem. Where we are, Dr. Chandrasekhar Pandey.
Yes, sir.
Please.
Sir, myself Dr. Chandrasekhar Pandey.
आपकी आवाज थोड़ी कम आ रही है, थोड़ा तेज बोलिए, प्लीज बी लाउड।
सर, ठीक हुई आवाज से?
ठीक है, बोलिए।
So myself Dr. C.S. Pandey. I am working as a scientist of horticulture at Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur.
मोबाइल एडवाइजरी हम लोग बना सकते हैं?
हेलो,
म्यूट करिए, बोलिए।
आवाज आ रही है?
अभी आ रही है, हाँ बताएं।
And sir, my field of specialization is fruit science, horticulture fruit science. Presently, I am involved in medicinal and aromatic plants as well as arid zone fruits. So in this project, I am involved in crop improvement program, especially medicinal plant like Gudmar, Satauri, Ashwagandha, Sarpagandha, and Betelgeuse. And in case of fruits, I am involved in survey and collection of custard apple, jamun, tamarind, and pear. So, sir, this training will be very useful to me in survey, collection, evaluation, conservation, and maintenance of the germplasm. So, by looking at this, I have joined this training program, sir. This training will be very useful to us.
The area of collaboration could be for you that we have a large number of accession in our
medicinal and aromatic plants.
Large number of accession in our gene bank. You can seek one of the exchange of the material, as Dr. Gautam told in their lecture. And another could be like we have also herbal garden. We have established so many accessions. You can visit and you can see how it can be done.
Okay, sir.
Okay. Thank you, Dr. Pandey. And next is Dr. Chandrakant P. Soregaon, US Dharwad. Dr. Chandrakant. If no, then Dr. Chinmay Mahapatra. Dr. Chinmay Mahapatra.
Yes, sir.
Please, madam. Please.
Very good afternoon to one and all present here. I am Dr. Chinmay Mahapatra. Currently, I'm serving as an assistant professor in Sri Sri University, Cuttack, in the Department of Plant Pathology. I'm also the head of the plant pathology here. So when it comes to this training program, when I did my PhD from Banaras Hindu University, where I was dealing with mostly powdery mildew of pea, and I was having 150 accessions like germplasms of pea, which was into my research. So there I was handling all this, and after joining Sir SSU here, I'm primarily into teaching, where my research is basically at a standstill position now. So this by having a knowledge on the present condition of the plant genetic resources and all, it will be I'll be more helpful, where I can have a clear cut idea about the recent researches going on, so that I can take my research further, especially on the powdery mildew aspect of the different diseases of plant.
Powdery mildew on which crop, ma'am?
Sir, pea, powdery mildew of pea.
So just I'm working on pulses just for information.
Okay.
So we
Okay.
can have something.
Yeah. Thank you, sir.
Okay, thank you. Nice to.
Sorry to interrupt.
हाँ madam बताइए.
Can I introduce? Because we have some.
Please, Dr. Kirtirani, please. Okay, please.
Yes, a warm greetings to everyone. My name is Kirti Rani. I'm working as a scientist and presently posted at a regional station, Jodhpur, NBPGR. As far my research experience is concerned, I have worked on the wheat, on genetics and molecular mapping of leaf rust resistance gene from IRA, Division of Genetics. And then I joined Directorate of Groundnut Research, Junagadh. So I have 4.5 years of experience on groundnut research experience. In groundnut, I worked on the management of groundnut genetic resources and trait discovery as a PI of the GRS unit and in charge of the GRS unit and PI of this project. As far breeding is concerned, then I worked on the breeding groundnut for earliness, fresh seed dormancy, and confectionery types. As a PI of this project, I have developed so many advanced lines, and many lines are in advanced trials. So after that, I got transferred to the research station Jodhpur, and it has been only 4 months me to join here. So I'm working forward to work with NBPGR and whatever is in mandate of NBPGR. So I'm willing to work with the great team of NBPGR. Thank you so much.
Glad to hear you, and of course you are an integral part of NBPGR. So I'll be also having opportunity to work with. Thank you. Thank you for introduction. And then Dr. Chirag Ashok Bhai Babaria, Junagadh.
Yes, sir.
Please.
Can I audible, sir?
थोड़ा आपकी voice थोड़ी सी slow है. Please bit louder.
Myself, Dr. C.A. Babaria, working as an assistant professor in Department of Seed Science and Technology, College of Agriculture, Junagadh Agriculture University, Junagadh. And I am a dual recognized. I am basically a plant breeder. For the genetics and plant breeding and seed science and technology, most of the PG teaching and PhD work we have related, and some groundnut and wheat pre-breeding work already we have carried out, and some idea of the program of the seed bank of in RKVI project we have proposed. That's why we have joining this training.
Okay, thank you, Dr. Chirag. And Dr. Chituri Manikya Mini from Jayashankar, Telangana University.
Morning, sir. This is Maniki Mini Chituri. I am presently working as a safflower breeder in agricultural research station concerned to Professor Jayashankar, Telangana State Agriculture University.
Safflower, you are working on safflower.
Yes, sir. I am a safflower breeder.
Are you part of the safflower network project?
Yes, sir. In all India coordinated research projects.
Okay, thank you.
Thank you, sir. Thank you, sir.
And the next is Dr. D. Malarviji, from TNU. Madam, please. Madam, please unmute yourself, please. Okay, thank you.
So is it audible, sir?
Yes, ma'am. Yes.
So good afternoon to all of you. First of all, I express my sincere thanks for selecting me for this training program. And myself, Malavili, and I'm working as professor in plant breeding and genetics in Department of Plant Genetic Resources, Tamil Nadu Agriculture University, Coimbatore. And my research work is related to varietal development in rice and red gram. And I'm having an externally funded project on green gram for development of bruchid resistance. And now I have been transferred to this Department of Plant Genetic Resources. And here we do the collections of traditional varieties in different crops, and also from the germplasm collections that are maintained in the different research stations of TNAU. And here recently we have established one cryo conservation unit also. So if any exposure is given towards this cryo preservation techniques, it would be good, sir.
Certainly, ma'am. It is a part of our training module, and there will be more, lecture and practical is there for cryo conservation and in vitro. So I assure you that you will be benefited with this. And anything you need, you can directly contact to TCC. You had one of the established division here, Tissue Culture Cryo Preservation Unit. You can call to the head and mail. In any way of communication, you can contact.
Okay, sir. Okay, sir. Thank you.
Thank you, madam.
Thank you, sir. Thank you.
And
Sir,
Doctor, please,
Excuse me, sir.
Dr. Lalu Prasad, isn't it?
Yes, sir.
You have raised the hand. You have raised the hand, please.
Yes. Just for introduction, sir. Actually, we are having parallel meeting here.
No issues, please.
So myself, Lalu Prasad Yadav, working at Central Horticulture Experiment Station, Godhra. It is the regional station of CIAH Bikaner, and working in ICR since 8 years. And presently, we are working on underutilized perennial vegetable crops in Coccinia, spine gourd, and drumstick. Besides this, we also working on improvement of tomato, bottle gourd, and pumpkin. And recently, we have added cucumber also. And we are working on collection, characterization, evaluation, and utilization of these crops. So our institute as well as station also is the part of the NAGS, National Active Germplasm System. So we are also maintaining the germplasm fruits as well as vegetable crops here, perennial horticulture crops. So the training will be really helpful for us to update our knowledge regarding utilization of the PGR. Thank you so much.
Okay, thank you.
Excuse me, sir. Sir, this is, I had to, sir, leave for a meeting. So may I be allowed to introduce myself?
Please.
Sir, myself, Dr. Tanmay Lalitandu Mahanti. Presently, I am working in Forest Biology and Tree Improvement Department of College of Forestry in Odisha University of Agriculture Technology. And we are working on the improvement of the perennial tree species, and particularly Acacia auriculiformis, Casuarina equisetifolia, Technograndis, Pterocarpus marsupium. These are some of the species we are working on it. And I'm in this department for the last 8 years. Now our basic thing, we are doing the things that we are selecting through selection, we are finding out some good genotypes and propagating and doing nursery studies and all those preliminary studies. And this training program, I think, will be helpful for carrying forward my research work in future, sir.
Okay, thank you. Dr. Padmaja.
Thank you, sir.
Thank you. Dr. Padmaja. If not, then Dr. Davara Dharmesh.
Excuse me, sir.
Who is around?
Myself, Dr. Udit Nandan Mishra.
Please.
Good afternoon, sir. Good afternoon to everyone. Am I audible, sir?
Yes, very much.
Yeah. So thank you for giving me this opportunity, and thanks for accepting my application to be a part of this training program. So myself, Dr. Udit Nandan Mishra. Presently, I'm serving as assistant professor in Sri Sri University, Cuttack, Odisha. So my specialization was in biochemistry and agricultural chemistry. So my expectation from this program would be a germplasm evaluation, then bioinformatics tool for trait evaluation, then genotyping. These are the aspects I'm looking forward to learn more from this particular program. Thank you, sir.
rest assured, we all will give you this kind of module what we're expecting from us.
Surely, sir. Thank you. Thank you, sir.
Yes, Dr. Kuldeep, myself, Dr. Taraknath Shah, Senior Scientist at ICR Directorate of Agriculture Research, Pune, Maharashtra. I am actually working on chrysanthemum, gladiolus, tuberose in flower crops. And in Directorate of Agriculture, we are having around 140 germplasm of chrysanthemum. And right now we are evaluating for mainly the cut flower, loose flower, and other traits. And recently, with the joining of some of our scientists from other discipline, they are also looking after other aspects like essential oil, then heat and stress, other things we are looking after. So my aim of joining this training was like that for evaluation, registration process in the NBPGR, and bringing the germplasm from outside the country also, if it is available in NBPGR or through other sources. So with this motive, I joined this training. Hopefully, I'll be getting some of the things I'm getting, all those things, and some more in future, in coming days also. I am expecting to learn more of the things in this regard. So thank you.
Sure. We'll be at your expectation, and definitely your registration guideline and what we have tried to accommodate everything what is needed, in your science. Thank you.
Sir, can I introduce, sir?
Please, ma'am, please.
Myself, Dr. S. Maheshwarama, working as a scientist at Akrepon Sorghum, RAR Palem, Nagar Karnool districts, Telangana, sir, from PJTSAU University. And initially, I was at teaching, sir, for 4 years, and got to research. And I am more interested in a collection of diverse lines of sorghum. For that purpose, I want to register those lines.
There are some interruptions from your side.
Yeah, sorghum, entire sorghum has been planted at Akola, no? So madam can visit during field day.
Yeah, madam.
Field day is going to be organized there at Akola.
Yes, madam.
Entire sorghum germplasm from gene bank is planted here. So you can visit during the field day.
Yes, we are planning for that one during the next.
Oh, that's great. Okay, thank you, madam.
Thank you, madam. Thank you, sir.
Can I introduce myself?
Yes, Dr. Kavita, please.
Yeah. I'm Kavita, associate professor working in the department of. Is it audible, sir?
And now it's fine.
Okay, sir. Sir, myself, I am Kavita, associate professor, Department of Seed Science and Technology, Tamil Nadu Agricultural University, Coimbatore. Previously, I worked in the Department of Plant Genetic Resources under Center for Plant Breeding and Genetics, and I handled a project on assessment of vigor and viability of germplasm accescents stored under ex situ conservation. And also I involved in viability testing and deposition of germplasm accescents in gene bank, along with Dr. Malarvili Madam, Professor, Department of Plant Breeding and Genetics. Thank you, sir.
Thank you, madam.
May I introduce? Hello, sir.
Who?
Sir,
hello. Ma'am, let me. That's why this is a problem. No, that's why let me intervene. If you have some urgency, then you can raise your hand. Otherwise, everybody is asking, then it will be difficult now. No? I understand, but please adhere to the instructions. It doesn't.
Sir, good afternoon, sir.
Who is there?
Sir, Dr. Padmaja, sir.
Please, madam. Yeah,
I'm Dr. D. Padmaja, working as a scientist at RARS Polasa Jagitya, Telangana.
Okay.
Sir, I'm Sir, 1 minute, sir. One sound problem.
What is your area of working, ma'am? Just let us know, and then we'll proceed.
Through this training program, we would like to register some of the lines, sir, because we are having some monostem and some water stress tolerant lines. We would like to register those lines, sir.
Okay, okay. But it will not be through this training, madam, na?
And we will. How we don't have a proper idea how we have to import some of the material which is suitable for the mechanical harvesting and those types, sir.
That Dr. Vandana Tyagi itself told that there are set procedures that will be applicable to all, not for just whether you are importing for high oil content or mechanical harvesting, the procedure will be the same. Okay.
Sir, actually, last class we have some idea regarding that. So this is.
So I hope you have got some exposure regarding the procedural thing with the lecture of Dr. Vandana Tyagi.
Yes, sir. Yes, sir.
And maybe we, Dr. Pragya Ranjan and Dr. S. K. Yadav will be taking a practical also how to fill the form. So please do attend in afternoon session.
Yeah, today afternoon, very important
Important.
session is there. So you will know all the procedures, how to import the germplasm from outside countries.
So please do attend in afternoon.
Please do attend that session.
Okay, sir. Thank you very much, sir.
Please unmute yourself first.
Sir, you are mute.
Okay, okay. Dr. D.K. Payasi, if you want to, because you are moving the field, please.
Okay. Myself, Dr. D.K. Payasi. I am scientist and in charge AIC, All India Coordinated Research Project on Lancet. As well as I am also coordinating DBT fund, sponsored network project on Lancet, where we are characterized already characterized 2,657 germplasm. NBP under the umbrella of NBPGR funded by Department of Biotechnology. I am also working on safflower for All India Coordinated Research Project. So these three different projects I am looking after. I belong to Jawaharlal Nehru Krishi Vishwavidyalaya, which is situated at Jabalpur and working under RARS Sagar. Current location is RARS Sagar. I have applied for this. I have applied for this training for career advancement and to procure the germplasm for economic trait of interest from the abroad. So how to proceed and other rule and regulation I have to know. That's why I applied for this.
Thank you, Dr. Sir, for introducing from the field. And you have taken 2,600 accession from NBPR Gene Bank. What else you need?
Yes. Yes, yes.
And maybe in other crops also. Maybe other crops also we can
and
share.
safflower.
Okay, okay. Safflower also there is a DBT project. If you will be the partner, then certainly you will be having these things, you know. But I don't know whether you are a partner in the safflower project also.
No. I am not actually. That will be the huge work, and we are suffering from the staff. That's why I am working on AICRP coordinated project. This is
no problem.
crop
You missed the bus last year, but again, if you are any program will be there, then definitely. Anyway, after developing the core set, you can ask NBPGR, then it will be provided. No problem. Thank you.
Yes, sir. Yes, sir. Sure.
Dr. Prajal Day.
Thank you very much, sir.
Dr. Prajal Day.
Good afternoon all. Good afternoon one and all. Good afternoon, sir. So myself, Dr. Prajal Day. I hail from one of the sister states of Northeast, that is Tripura. I am working currently here at Sri University Faculty of Agriculture, and I am from the discipline of crop physiology. My area of research is on abiotic stress, and today's morning talk has been quite helpful for me. And also Dr. Vandana Tyagi's talk, what has been talked today, what Madam has discussed. I have worked on germplasms related to low light stress tolerance in rice. So this today's lecture and the lectures what has gone through will definitely help me to screen and isolate more varieties regarding to abiotic stress tolerance, and it will have definitely an impact on my research area of work in the coming days.
Okay, thank you, Dr. Prajal. Dr. Vikas Pawar.
Am I audible, sir?
Yeah.
I am Dr. Vikas Pawar, working as an assistant professor agricultural botany in MPKV Rahuri University. I have 24 years of experience of research. Out of that, 22 year nearly in per millet, and I have registered near about the 3 hybrid with its parent and 1 variety with NBPGR. And recently I have joined in teaching MSc as well as BSc Agri, and I joined this training with a motive for utilization of PGR and evaluation of germplasm, and especially for registration of germplasm, which were collected collected with the help of student in RAWE program of undergraduate student. And I am very thankful for selecting me for this training.
Thank you, sir. Thank you. Thank you for your this talk. Dr. Monu.
Good afternoon, all. Myself, Monu Kumar. I did my master's from IRI New Delhi in genetics, and there Dr. Kuldeep was my bestmate, and Dr. Jyoti Madam taught my some of the lectures. So it is good to be part of this training program. And I joined my ARS in 2014 at Krajab Barakpur. And 2 years back, I transferred in the new campus of IRI, that is located in the Jharkhand, IRI Jharkhand. And right now I'm working on wheat and pigeon pea improvement programs. And I am also looking after the UG and teaching here and just started now. So that's all about.
Thank you, Manu. It is always good to listen to you. Thank you. And Dr. P. Gonia Nayak. Dr. Nayak from PGTSAU? No. Dr. Chandrakant B. Soregaon, please.
Good morning, sir.
Yes, thank you.
May I audible to you, sir?
Dr. Chandrakant, please.
Myself,
May I audible to you,
B.
sir?
Soregaon. I am working as assistant professor at College of Agriculture, Vijaypur, which comes under University of Agriculture Sciences, Dharwad. I am basically involved in teaching UG as well as PG courses, and I am involved in research on pigeon pea breeding, especially working on earliness aspect. And I am also evaluating some of the mini core collections from ICRISAT in pigeon pea. And this training course will help me to have these all germplasm evaluation, and which can be used in crossing program, and that basically helps in teaching as well as research aspects. Thank you.
Okay, thank you. Just for information, under CRP agrobiodiversity program, we have characterized around 10,000 accession of pigeon pea. And hopefully by the end of this year, we'll be having entire set of data. So that will be useful for you. So maybe
Okay.
we will discuss later, no problem.
Okay, thank you.
Okay, thank you.
Good morning, sir.
Please, Dr. Nayak.
This is Dr. Nayak, sir, working as scientist in rice breeding, sir, at RRS Jagitial, Professor Jayashankar, Telangana State Agriculture University. Earlier 4.5 years, I have worked in AICRP and groundnut breeding, sir. And from last 5 years onwards, I am in rice breeding, sir. Through this training program, I will know the characterization of germplasm and their utilization in rice breeding program, sir. Presently, I am working on development of long slender rice varieties resistant to BPH and blast, sir.
Okay, Dr. Nayak, but just for information, we have Dr. R.K. Gautam with us, and he is a rice expert throughout his career. He has worked on rice. So maybe, sir, you can.
Yes, Dr. Nayak, it's happy to know that you are working on rice in Telangana.
Yes, sir. Yes, sir. Good morning, sir.
Good morning. Which place in Telangana?
RRS Jagitial, sir. Jagitial Center.
Okay, so there are two important institutions in your state there. One is Maruteru. I think you must have visited Maruteru station. That's the oldest, probably a very old and very famous station for rice. Maruteru.
Yes, yes, sir. Yes, sir.
And since our Indian Institute of Rice Research is there in Hyderabad.
Yes, sir.
So are you attending the ACRIP workshops every year?
Yes, sir. Yes, sir. Attending, sir.
Very good. Very good. So as far as this BPH is concerned, there is Dr. Padmavati. She worked on these BPH resistant genes in rice. So you can see her work also, BPH resistance genes. Okay.
Okay, okay, sir. For Kharif 2020, we have sent 13 BPH tolerant genotypes for screening, sir, in national level.
What is the main disease in your area? Disease.
Blast, sir. Blast and BLB is the major disease.
Okay.
Almidge and BPH are the major pests, sir.
Okay.
Dr. Nayak, just suggestion from my side, be in touch with Dr. Arti Gautam, then you will be more benefited. Okay.
Okay, okay, sir.
Okay.
Thank you, Dr. Nayak.
Okay, sir.
And anyone, whoever is online, please introduce one by one because we have distracted from, Dr. Kaushik Panigrahi, please.
Yeah, sir. Good morning to all of you. This is Dr. Kaushik Kumar Panigrahi from Odisha University of Agriculture and Technology. Currently, I am posted in the sub-campus of OUAT and dedicated to 100% teaching. I am involved in teaching and a little bit of research work we are doing in pulse crop here. So this training, I have attended this training to gain some knowledge because I have not yet started any research work, means like a agri project or any other project. Since my joining, I am in teaching. So in future, if I'll get chance to work in any agri project, this knowledge, what I gained from this training will be beneficial for me.
Okay, thank you, Dr. Kaushik. Dr. Piyusha Singh.
Good afternoon, sir. Myself, Dr. Piyusha Singh. I am working as assistant professor in Department Genetics and Plant Breeding in Acharya Narendra Dev University of Agriculture and Technology. And I am working in teaching also as well as research also. Fodder crops are the main, my work is on fodder crops. And this training is very beneficial and I want to know how to get the IC number of different germplasm. So I joined this training.
What is the research area, madam?
Fodder crop, forage.
Forage, what? Legumes, grasses?
Sir, oats.
Okay, thank you. Thank you.
Thank you, sir.
And Dr. Vishnu Gite, please. Please unmute yourself.
Sir, good afternoon, sir.
Please.
Myself, Vishnu Gite. I am working as a mullar breeding in ECRIP programs and also in pigeon pea breeding as a breeder for the 8 years. I released the 4 varieties of pigeon pea and 1 variety of mug pea. I also work as a co-PI in 2 ICR sanction project, one on pigeon pea, that is the CRP on Agri Technology project. Second one with NVPGR collaboration, that is the characterization and evaluation of genetic resources for genetic enhancement of minor pulses. Under this project, we evaluate near about 4,100 germplasm lines of moved in 2 consecutive years, and during the last years, we evaluate 425 cowpea germplasms in collaboration with NVPGR programs. I also joined the one exploration program with the NVPGR Akola substation for exploration of wild species of pigeon pea in Vidarbha regions. Thank you, sir.
Thank you, Dr. Gite. Dr. Anuj Kumar Singh, please. Dr. Anuj.
Good afternoon, sir.
Please.
I am Anuj Kumar Singh. I am working as assistant professor in the discipline of plant physiology at SD Agriculture University. Presently, we are working on evolution of sesame genotype for drought and salinity tolerance. We are part of the network project at NVPGR, which is coordinated as NVPGR.
Okay, that's great. Dr. R.C. Meena, please.
To all of you, myself, Dr. R.C. Meena, as an assistant professor in plant physiology, sir, ECRIP on Power Village, project coordinating unit, Jodhpur. I did a PhD from Botany of Department, University of Rajasthan, Jaipur, sir. Basically, I am working on in project coordinating unit ECRIP on Power Millet in Jodhpur. I'm working in the area of the abiotic stress. I mean, also screening the permanent genotypes for to develop the drought tolerant and high temperature hybrids. Thank you, sir.
Okay, thank you to know about you. And who else is
Sir.
there? Please introduce.
Sir, myself Harman Tapati, sir. Sir, morning, sir.
थोड़ा loud बोलिए, please bit louder.
Sir, hello.
Yeah, hearing. You can.
Sir, myself, Dr. Harman Tapati, sir, working as assistant professor of seed science and technology, University of Agricultural Sciences, Raichur, sir.
Okay, which crop you are working?
Sir, I am working as a farm superintendent and teaching, sir. Farm superintendent in the major our station was millet seed production like foxtail millet, proso millet, little millet, brown top millet, and ragi, sir. I
Okay,
want to
that's
join
good.
this program because of how to know IPR issues and dust testing. I want to know, so that's why I am joined to this program, sir.
Okay, okay. You will get during the lectures, all your queries
Yes,
will be answered.
Ma'am, can I?
Yeah,
Dr.
yeah, sure.
Vajanti, Dr. Vajanti, raise hand, please. Dr.
Sir,
Vajanti.
am I audible?
Yes, madam. Yes, very much.
Yeah, good afternoon, everyone. Myself, Dr. Vajanti. I am a faculty from Kerala Agriculture University. And my present assignment include both teaching and research. Teaching include UG, PG, and PhD courses. And in research, I have an ongoing CERB project on rice salinity tolerance. And recently we are funded with an RKVY project on seed. So we are trying to collect germplasm focusing Northern Kerala, including biodiversity hotspot Wayanad. So from this training, I am expecting some important exploration aspects of germplasm, how to register it with NBPGR, and also how to collaborate with other institutes by considering Biodiversity Act and all. Thank you.
Just a minute, Dr. Kuldeep.
Please.
Yes,
Yes, madam, you are working in Waitila. Where are you working?
No, I am in College of Agriculture, Padanakad, Kasargod, sir.
Kasargod.
Yes.
So it's a coastal area, coastal.
Yes, sir. We have coastal saline area, sir, here.
Oh, very good, very good.
Yes,
so you must be, attending rice workshops also every year.
Right now, I am not a part of ECRIT, sir.
Okay, since how many years you are working in rice?
From last 4 years, sir.
Okay, okay. Then have you heard about one institute on working on soil salinity in Karnal?
Yes, sir. I am aware of Krishnamurthy, sir, and team, sir.
Oh, very good, very good. Then it's very nice. All the best. Okay.
Okay, thank you, sir.
Dr. Shashikala.
Hello, good afternoon, sir.
Please.
Myself, Dr. Shashikala Kolkar. I'm working as an assistant professor at College of Agriculture, Shimoga, TSN UHS, Shimoga University. Basically, I'm involved in teaching as well as research, sir. So this training will help me to enhance my knowledge regarding registration of the varieties, even dust testing and all. Thank you, sir, for giving me this opportunity.
Thank you, ma'am. Dr. What next? Dr. G. Tanvendan. Unmute yourself, Dr. Sir. आपकी voice नहीं आ रही. You're not. Now it's clear.
My name is Dr. G. Dhanavendan. I am basically agricultural entomology. So since 7 years, I am working in mulberry blossom collection and conservation and evaluation at Hosur, Tamil Nadu. So my aim is here, I want to learn especially for the trait-specific germplasm, how to register it. Since you know that mulberry is a multi-purpose, you can use foliage and fruits and more value-added product also. So that's why I want to learn how to register the trait-specific mulberry germplasm, because it is a center for the mulberry germplasm NAC center. It is recognized by the NBPGR. So recently we supplied for this fruit taxation to Sir Morning, that agroforestry Magapatra sir, and we recently collaborated with the Koloriang survey also, Dr. Subhash Chandar and Narendra Nagi. So we are expecting how to registration for that. I joined, sir. And thank you for giving an opportunity.
Thank you. Dr. Vinu Aher from JU. Unmute करिए.
Sir.
Dr. Vinu, please. Voice नहीं आ रही, Dr. साहब. Till then, please check yourself. Till then, we can take somebody else. Dr. Sujata, madam, please. Dr. Sujata, please.
Yes. Good afternoon, everybody. Am I audible, sir?
Yes, ma'am. Yes, very much.
Actually, I am from Agriculture Research. I'm Sujata from Agriculture Research Station, Tandur, which comes under Professor Jaishankar, Telangana State Agriculture University. I'm a sorghum breeder, sir. Since 9 years, I'm working in this field. And my expectations are to understand the procedures well for germplasm exchange with partners, and also germplasm registration, and also to improve my own breeding efficiency with the help of PGR tools. That is understanding the diversity very well, especially in Rabi sorghum germplasm, where the diversity is very much limited. So to understand the best, to identify the best lines, use them in crossing, especially when the resources are very much limited. And that's all. Thank you.
As Dr. Jyoti told that there is a large scale experimentation of more than 25,000 accession are going to be held in Akola. Please do visit the center, and then I hope that you will not say the diversity is limited when you will visit.
Okay.
Thank you, ma'am. Thank you.
Can I introduce?
Dr. Somveer Singh, please.
Sir, I am Dr. Somveer Singh. I am working as wheat breeder in Department of Genetic and Plant Breeding at CSA University of Kanpur from last 10 years. I am also working as wheat breeder, sir. We developed 2 varieties of wheat and 1 variety of barley. 2 varieties, namely wheat of varieties are K13, 17 and K16, and barley varieties KV14, 25. I hope this training will be beneficial for me, sir. Thank you.
There are 2 gains for you. One is I belongs to Kanpur. First thing.
Okay.
Second thing, you are wheat breeder. Then Dr. Jyoti Kumar is here.
Yes.
So
Dr.
she is
Somveer, welcome to this training.
Thank you, madam. Thank you.
Yeah.
Good afternoon.
So come to field day if we plan this year. You have come earlier, I remember.
Yeah, sir. I meet 2 times, madam, in workshop, wheat and barley, and
Madam is key person.
Yes.
Yeah, I
every
year.
I know very well, sir.
Okay, thank you.
So
Dr. Bharati,
do
madam.
visit the field day when we are plan. Maybe in March we will be planning mid-March.
I already visited 2 years back.
Yeah. You came that year. I remember very well.
Okay, thank you, Dr. Somveer Singh. Please, Dr. Y. Bharati, madam, please. Dr. Bharati from PGTS.
Good afternoon. Am I audible, sir?
Yes, ma'am. Yes.
Myself, Bharati, from Assistant Professor from PGTSAU, Professor Jaishankar, Telangana State Agriculture University.
Ma'am, please bit louder.
Yeah, okay.
Come
Myself,
close.
Bharati, from PGTSAU, Assistant Professor. Presently, I am teaching students of Department of Seed Science and Technology, and also we are doing research also. Basically, we are doing characterization of soybean germplasm for seed characters. Case for seed longevity, we are looking for.
Okay, just ma'am, for information, Dr. R.K. Gautam, he is taking care of the soybean here in NBPGR. Recently, my sir has taken a large initiative, and we have characterized the entire set of soybean accession at 2 places. Maybe sir will intervene in between. Sir, please.
Yes, since she is working on soybean and germplasm is the issue. So, madam, I want to just update you, and I also mentioned in my presentation that we characterize the entire set of our gene bank collection plus accessions from Indore, and the total number was around 8,000 accessions. These were characterized at Bangalore actually by GKVK, and we were part of that field day also, and many neighboring breeders, soybean breeders had come there. So for further information, you can contact either ISR soybean or GKVK also. Okay. All the best.
Okay. Thank you, madam. Dr. Veenu.
Sir, can I introduce Dr. K. Gupta?
Yes. कौन है? कहाँ से?
Dr. K. Gupta, sir.
Please, आप करिए, हाँ, please.
Okay, sir. Thank you very much, sir. Sir, myself, Dr. K. Gupta, from JNKV Jabalpur. I am working as a scientist since last 13 years. And then I am working in so many screening of germplasm, including bi-germplasm. And I have one project, a collaborative project with NVPGR, identification of tolerant line against sesame, phyllodes and macropulmonary root rot disease. And we have also screening of large scale germplasm, utilization, characterization, and further conservation. And I also have some teaching program also. And this program we have useful because we have characterized the sesame germplasm line and bi-germplasm, sesame and Niger both. So further improvement of our work. Thank you very much, sir.
Hello, sir.
Sir.
Yes, you can introduce yourself, Dr. Patel.
Yes, audible, sir, madam.
Yes, you are very much audible.
My name is Shashikant Patel. I am working on AICRP project of wheat and barley at a wheat research station, Bijapur, Gujarat.
Okay.
I am looking the AICRP on wheat and barley during the last 4 years, and we developed the 2 state variety and the 1 national variety GW513. Also, we are registered the 4 genetic stock for various state at NDPGR for the high protein content and various agronomical trait. Also, I am looking after the wheat breeding. The targeted trait is for terminal wheat stress, operation, durable rust resistant variety at a Gujarat state.
Okay, great to know that. So, Dr. Patel, do you know that NVPGR has developed wheat core, wheat mini core as well as barley core? So, if you wish to work on that, so means you can get intent for that, and you can work for
Also,
your interest.
during 2 years ago, we are also collecting the material for the field day at NPPGR.
Okay. Great. So now barley core is also available. So means if you wish, you can take up that also. Okay. So great to hear from you.
Okay.
Reena Mehra.
Good afternoon, all. Am I audible? Hello, am I audible?
Yes.
Good afternoon, all. I am Dr. Reena Mehra, working as a lentil breeder in Ikada FLRP, which is situated in Madhya Pradesh, Amraha. And I'm having more than 10 years experience in lentil breeding program. And I'm very thankful to you and PGR for selecting me for this training program. I'm so sorry, I'm having severe cold, so that's why I cannot talk much. So
No
thank you
issue.
so much.
Okay, thank you, Reena. Okay.
Hello.
Yes, Dr. Ahir. Yeah, continue. Introduce yourself.
Yes, ma'am. Am I audible?
Yes, very much audible.
Okay. Hello, good noon to all of you. I am working as an assistant research scientist at the Agriculture Research Station, Mohua, Junagadh, Gujarat. Currently, I am working on the coconut varieties to develop a quality seedling of different coconut varieties. And I'm also involved in different teaching and extension activities of our district. And now in past, I have also worked on different seed production program like groundnut, chickpea, sesame, onion, and castor. And to gain some knowledge about the different processes for the sale of the germplasm and how to import and export the quality of germplasm. That is why I'm joined to this training. And thank you very much to all of you. Yes.
Okay. Thank you, Dr. Ahir. So you have some lecture on coastal diversity, germplasm. So you can get some things of your interest from there.
Ma'am, can
Yes.
Yes, sure.
Ma'am, good morning to all of you. Ma'am, my name is Mohammad Raza from ICAR Kazri Arrest Leh. And I am working as a senior technical assistant since 2015.
Okay.
And this program is most benefit for me, how to collect the local germplasm in here And I am also looking for a J project also in here
Okay.
in Leh
Okay,
district.
that's good.
And we have set up a local seed bank in here, namely Stagmo village. And we have in here Ladakh, we have two district here, Leh and Kargil. We have one village in here in Leh and two village in the Kargil under J project.
Okay, good to know about your work. Thank you.
Yes.
Dr. Raja.
Thank you so much, ma'am.
Okay.
Hello.
And thank you so much, sir.
Hello.
Kuldeep sir. Thank you so much.
Hello.
Yeah, who is
May I?
online, please?
I'm Dr. Sunil Gawale from Ratnagiri, Maharashtra.
Okay. Yeah.
Myself, Dr. Sunil Gawale. I'm a research officer, assistant professor in the regional coconut research station, Bhate, Ratnagiri.
बताइए
Now I'm working as a plant breeder as a coconut research station, Bhate. There are 11 experiment going on about crop improvement, especially in genetic resources, that conservation, evaluation, and utilization of plant genetic resources, particularly in coconut and spices also. So this training will helpful to me. Hello. Hello. Hello, madam. Hello.
सुन रही
This training will helpful to me for future.
Yeah, thank you.
Okay.
Madam, can I help introduce me?
Yes, sure.
Myself, Dr. Gopal Shukla, working as an assistant professor in Department of Forestry at Uttar Banga Krishi Vishwavidyalaya, Pundibari, Coach Bihar since 2014. My area of interest is medicinal and aromatic plant, and I joined this training particularly to know how to register the germplasm in your institute. And this course is also helpful for me to my teaching, particularly in UG and TT. Some of the course related to forest genetic resource. So after this training program, I think I will be teaching my student in a better way. Thank you, madam.
So, Dr. Sukla, we have a specific lecture on forest genetic resources, and as well as for registration guidelines also we have separate lecture. So
okay,
you'll know everything from there. Okay, thank you.
Thank you.
Hello, ma'am. Hello, ma'am.
Dr. Savita, yeah, please. Dr. Savita, please.
Yes, ma'am. So good afternoon, one and all. My name is Savita SK. I'm working as an assistant professor, Agriculture College, Vijaypur. It is a sub-campus of US Dharwad. Recently, 1 year I joined as an assistant professor, only handling the UG courses, that is undergraduate courses. I'm joined this course to gain or widen the knowledge about the germplasm resources, so that in future, if I get the chance to work with, so I can apply this knowledge. So thank you so much, madam.
Thank you, Dr. Savita.
Hello, ma'am.
How about you?
Hello,
Dr.
ma'am.
Binod. Dr. Binod.
Yes, ma'am.
Yes, please.
Good afternoon, ma'am. Myself, Dr. Binod Kumar, Assistant Professor, College of Agriculture, Pawar Kheda, Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur. Mein. I am associated in teaching, and I am also working in wheat crop improvement, madam, AICRP in wheat Pawar Kheda. And we have 6,000 germplasm, and we want to characterize them. This training is very helpful to us, ma'am.
Okay, nice to know about you. We are also conducting our trial at with Dr. KK Misra and Dr.
Yes. KK Misra is our PI, madam, project in charge, and our breeder, madam.
Yeah, so we are taking up heat and drought trial at as one of the center at Pawar Kheda. Okay, so
okay.
Thank you very much. Nice to know about you.
Okay.
May I, ma'am? Hello, ma'am.
Yes, Seetal, Dr. Seetal, please.
Yes, ma'am. This is Sheetal Patel from Navsari Agriculture University. I'm presently working with the rice crop and groundnut crop. So main aim of to join this training is to gain the information regarding the germplasm conservation and collection of a particular groundnut crop for objective of having earliness.
Okay.
So ma'am, one more question, that is there a field day for groundnut crop?
No, NBPGR Institute is not conducting field day on groundnut crop, but you may visit that ICR Institute, Junagadh. So
Yeah.
that place you can visit.
Okay, ma'am.
Okay.
Okay,
Thank you, ma'am.
Thank you.
Yeah, may I?
Hello.
Yes, Dr. Mohammed. Yeah.
Makbul.
बताइए.
Yeah, good afternoon to everyone present here in this online training program.
Very good
Myself, Dr. Mohammed Makbul Rathar, working as assistant professor in the Division of Forest Biology and Tree Improvement, Faculty of Forestry in Skost, Kashmir. Mainly, our work is in the field of teaching, teaching of just UG, PG, and PhD students. And regarding this research work, we are trying to improve our this indigenous species like poplars and silex for their improvement programs. Thank you.
Okay, nice to know about Dr. Makbul.
Thank you so much.
Thank you.
Hello, ma'am.
Yeah, so anyone is left now?
Hello, ma'am.
Yeah, please.
Good afternoon.
Yeah, Rosni. Yeah, please.
Good afternoon, ma'am. Myself, Roshni Sahu. At present, I'm working in project coordinating unit Sesame Nizer, JNKVB Jabalpur. Here in our project, ma'am, we are characterizing about 6,000 germplasm of sesame and around 2,000 germplasm of Nizer. The present training is very useful for us, for me to characterize, and we are also dealing with the quality parameters, ma'am. So like mineral composition and fatty acid profile. So is there any lecture related to this, ma'am?
Yes, we have a specific day on quality also. So we are having the quality practical also as well as lecture also.
Okay.
So you'll get those things. And in CSEM and NISER, we are developing core also. So in future, you can get that core for your specific purposes. Sir, आप कुछ कहना चाह रहे हैं?
Yes. Are you part of our DBT network project also on characterization?
Yes, sir. In DBT funded project, sir.
You are already partner in that project.
Yes, sir.
Oh, very good. Then you are then you are in touch with the latest developments. Okay.
Yes.
Thank you.
Okay. Thank you, sir.
Thank you.
Hello.
Yes, Dr. Pallavi. Yeah, please.
Hello, good afternoon, ma'am. I'm Dr. Pallavi, working as assistant professor in the Department of Seed Science and Technology from PJTSAU. I'm basically involved in teaching, and we do some research even in seed technology. So I've joined this training program mainly to gain some knowledge on germplasm registration and storage, which may be useful for teaching our students and also maybe in our future research programs. Thank you for giving us this opportunity.
Okay, thank you, Dr. Pallavi. Nice to know about you. Okay, thank you.
Madam.
हाँ.
Hello.
हाँ, आपका नाम नहीं आ रहा है. हाँ, बताइए.
Madam.
Yeah, बताइए सुन रही.
Myself, Dr. M. Ramesh, madam.
Okay.
Working as assistant professor, Department of Seeds and Technology, SRTC at Rajendra Nagar, madam, PJTSU.
PJTSU, okay, great.
पहले मैं 6 साल पल्सेस में काम किया मैडम.
Okay, good.
वो पल्सेस में स्पेशली रेड ग्राम and another horse gram. Lot of germplasm, nearly 500 germplasm in horse gram. I almost 150-200 evaluated, madam.
Okay, nice.
Now working here SRTC, madam, as well as teaching and research, madam.
Okay.
Earlier in PG teaching, madam, there is a in situ, ex situ conservation of germplasm. One course is there, madam.
Okay.
But now recently 1 year back they changed BASM, broad agriculture system. Now the course is not there, but the course is very utilized for me, madam, because of very in depth of germplasm exploitation, characterization, how evaluation is a very in depth knowledge is very useful for me. Future in guiding students and in my own knowledge increasing, madam. I am giving this opportunity very thankful to your team, madam, and especially thank you, madam.
Okay, thank you very much. Certainly, so our training will fulfill your expectations. Thank you.
Thank you, madam.
Who is next? Whether anyone is left?
Hello, madam.
Yeah.
Hello, ma'am. I'm Mrs. Prabhasmita Satpathy. Good afternoon to everyone.
Yeah, very good
I'm
afternoon.
working as assistant professor in College of Agriculture, Chiplima, which is a sub-campus of OUAT, Odisha University of Agriculture and Technology. So, sir, madam, basically, I'm into teaching for the last 7 years.
Okay.
Along with this, I'm also guiding postgraduate students. I have also worked on abiotic stress. Like I've worked with different rice germplasms under complete submersion stress, followed by long-term water stagnation in the field. So this training will help me to explore the field of germplasm conservation so that I can work more efficiently in the field of abiotic stress physiology in collaboration with this. So thank you, madam.
Yeah, sure. Our training is focused mainly on abiotic stresses, phenotyping for abiotic stresses. So you will certainly get very good knowledge out of it. Okay, thank you. Thank
Sure,
you.
sure. Sure, ma'am. Sure, ma'am. Thank you, ma'am. Thank you, ma'am.
Hello, good afternoon. May I
Yes.
introduce myself?
Yes, yours, Dr. Goris.
This is Dr. Gaurish Karanjalkar from Goa College of Agriculture, Goa. And basically, I did my PhD on molecular aspects of mango peel color from IHR Bangalore. And presently, I'm working on some of the underutilized fruit crops. And I was interested in knowing about GI tagging, seed bank also we wanted to set up because ours is a new college. And this training will be helpful for me in knowing all these type of aspects. Thank you.
Okay, thank you. Thank you, Dr. Goris.
Hello.
Yes, ma'am. Please.
Yeah, myself, Dr. Dipti Singh, and I have completed my PhD from GV Pant University of Agriculture and Technology. And recently, I'm serving as an assistant professor in the Department of Vegetable and Flowers at Bihar Agriculture University. And right now, I have just compiled all my projects regarding dry flowers. And in PhD, I was continuing with the marigold breeding, and soon I'll be submitting a project on that. So I think this training would be very beneficial for me for the procurement of different flower crops, as well as to fulfill my research, teaching, and extension activities. So thank you very much.
Certainly, ma'am. So today's lecture, Dr. Pragya will be taking up. So she is dealing with ornamental crops. So if you have some specific query, you can ask her. Okay. Okay. Thank you, ma'am. So hope now all are done. All have introduced themselves.
Good afternoon, ma'am.
Okay, बताइए.
Myself, Naresh Kumar, ma'am.
Naresh.
I am working as assistant professor now in KVK. Pathankot.
Where is video?
Oh, sorry, ma'am. Sorry. I am just.
चलिए अब ठीक है. Okay.
जी, ma'am. How are you, ma'am?
Fine. बताइए.
Yes, ma'am. Presently, I am working as assistant professor के कृषि विज्ञान केंद्र पठानकोट. Before that, I was in research, and presently I am involving in extension activities, and I want to explore my knowledge. How can I benefit the farmers, those who are cultivated the local varieties in the areas, and for my career advancement scheme. For my career advancement, so one training is mandatory for us. So that's why I am joining this course. Okay. And before that, I have also got the opportunity and learned a lot from you. So, so that's why I am interested to join this course.
Okay, thank you, Naresh.
Thank you, ma'am.
Nice to know about you. Okay. So
thank you, ma'am.
Now I think all are done. So shall we close the session, introductory session?
Yes, madam.
Hello.
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 Yeah, Dr. Mahesh.
Hello. जी मैम.
Okay.
जी मैम. First of all of you, very good afternoon. Myself,
very
Mahesh
good.
German, as a senior technical officer. I am working in ECRIP on wheat, JDRS for Kheda, JNK, Jabalpur. I want to learn in this training about germ plant collection, conservation, and utilization, and how to conserve new germ plant line in our center.
Okay, thank you. Thank you, Dr. Mahesh. You'll certainly get your expectations from the training. Okay.
Thank you.
Okay. I'm seeing one raised hand, Dr. Sasikala. I think the raised hand is by mistake or something. So now.
Hello, good afternoon, ma'am. I'm Vimla from Dr. Y.S. Horticulture University, working as a scientist at Horticulture Research Station in Anantapur. It is under ECRIP arid zone fruit crops. So we are working on germplasm collection, evaluation, and maintenance of bear, custard apple, tamarind, onla, ma'am. So I want to know the information and how to collect so new germplasm and the generation of IC numbers to that number, to that best evaluating germplasms. So this training will help on this on that aspect, ma'am.
Okay,
Thank you.
Thank you, Dr. Bimla. Nice to know
Thank you.
about you. Okay.
Thank you, ma'am.
So who is next? So I think now everyone is done. Maybe some are missing because in attendance total persons were not, all the persons were not present. So maybe we'll take up later, whoever is absent.
Good afternoon, ma'am.
हाँ आप भी बाकी हैं. हाँ या
हाँ जी,
बताइए
ma'am.
बताइए tell.
Hello, ma'am.
Yeah, I am hearing. Yeah, please tell. Please
Good afternoon,
introduce.
ma'am. Myself, Sunil. I'm working as assistant professor in Kerala Agriculture University. I'm recently joined Kerala Agriculture University. It's been only 4 months I have joined. I'm assigned only courses, UG courses, PG and PhD courses. Only as per now, I'm only having teaching. I'm not assigned any research work because I just recently joined. And this program and training will be very useful for me so that I can shine in my academics as well as research in research programs. Thank you so much for accepting my request, ma'am. Thank you.
Okay, thank you, Sunil.
Thank you, ma'am.
So I think now all are done. So I'm closing the session for today, introductory session. So the next class will be at 2:30. So please join at 2:30. It is a very good lecture by Dr. Pragya and Dr. S.K. Yadav regarding how to import the germplasm, from where to import. So all the procedures, so they'll be taking up import permit, preparation of import permit, how the online form will be filled up. So please attend. I request everyone to attend this specific lecture, very important. Okay, so please join at 2:30.
Thank you, ma'am.
Okay, thank you. Thank you, one and all.
and what are the procedures of germplasm exchange. It is highly informative, actually, this session. Still 45 candidates are joined so far. Okay, then let me start this session, and welcome back. And now I am introducing the current speaker, next speaker. This is our first speaker, Dr. S.K. Yadav, working as a principal scientist, germplasm exchange and policy unit. And mainly he is involved in the exchange of PGR in the vegetable crops, plantation crops, bulbous crops and other tuber crops. And he facilitated the introduction of several trait-specific exotic germplasm of horticulture crops from various countries, which are using for the crop improvement programs. And he, as a team leader, he is very active in conducting exploration programs also. And more than 45 multi-crop explorations conducted and collected nearly 3,790 accessions. And also he involved in characterization and evaluation of PGR in vegetable crops, exclusively tomato, brinjal, bottle gourd, sponge gourd, and ridge gourd. And besides, he worked in the research work of, he developed a core set of 181 accessions in brinjal. And he is a fellow of Indian Society of Seed Technology, Indian Society of Vegetable Science, and Indian Society of Plant Genetic Resources. And he is a reviewer in several reputed journals. And genetic stocks also, he registered 7 genetic stocks in vegetable crops and a buckwheat for novel traits. And he published more than 54 research papers and other books, book chapters, bulletins, technical proceedings, and some Hindi popular articles also there. And next speaker is Dr. Pragya. She is working as a principal scientist, horticulture in the division of germplasm exchange and policy unit. And she involved in the management of germplasm of horticulture crops. And she facilitated the introduction of several trait-specific exotic germplasm of horticulture crops. And she involved in exchange of genetic resources of fruit crops, leafy vegetables, and cucurbitaceous vegetable crops and ornamental crops. And she is, we can say proudly, she is a very high expertise and vast contributions in cucurbitaceous vegetables. And characterization and evaluation of genetic resources of vegetable crops. And she identified a novel source of carotenoid in orange flesh cucumber from northeastern India. And she registered 3 unique germplasm of cucumber for use in the breeding programs. And she developed 7 varieties, out of which 3 are Indian spinach, 2 are chinopodium, leafy amaranth 1 variety, and garlic is a 1 variety. And she is a reviewer in several reputed journals, and she published more than 36 papers and popular articles and book chapters and other technical articles, proceedings also there. And sir, we welcome you both, sir and madam. And sir, these are the participants of 84 candidates, which are from different organizations, 18 different organizations. And please kindly deliver our lecture and what are the procedures of germplasm exchange and how to get the procure of material. And a lot of research fellows having doubts of regarding the basic things of this germplasm exchange. Even we also don't know some way how to get the material and what are the forms are available and what is the procedure. And please, sir, kindly start your presentation. Ma'am, share screen, ma'am.
Thank you, Ravi.
Okay, thank you, Dr. Ravi.
Thank you, sir.
Let's just share our screen.
Yes.
Is it okay?
Ma'am, presentation mode.
Ma'am, presentation mode, ma'am. Yes, ma'am. Yes, yes. Please continue, ma'am.
Yes. Good afternoon, everyone. At the outset, I would like to thank the organizers for giving us the opportunity to conduct this practical session on application for import permit, material transfer agreement, procedures for exchange of germplasm. The session will be conducted jointly by me and Dr. S.K. Yadav. So let's start. As we all know that biodiversity in general and plant genetic resources in particular are at the core of the wealth of any nation. So we'll quickly define plant genetic resources as the genetic material of plant origin of actual or potential value in the form of seed, vegetative propagule, tissue, cell, pollen, DNA molecule, etc., containing the functional unit of heredity that can be utilized in crop improvement. And all these comes under plant genetic resources. These are land races, wild relatives, transgenics, farmers varieties, even release varieties. Everything is PGR only. So, germplasm wealth of any nation is composed of its indigenous natural diversity as well as introduced diversity, which is shared across national boundaries. So despite enormous genetic diversity, no country is completely independent for its germplasm requirement and it always looks for new germplasm sources from other countries. So that leads to germplasm exchange between those countries. And it is not a new activity. It took place since times immemorial. Crop plants have traveled into new areas from the centers of diversity through travelers, settlers, traders, invaders, explorers, etc. And all the countries are interdependent for their germplasm requirement. To emphasize more upon the exchange of germplasm, Thomas Jefferson has told that the greatest service which can be rendered to any country is to add a useful plant to its culture. So sharing and exchange of germplasm within and beyond national boundaries has generated opportunities to enrich the plant wealth of the country, and a diffusion of plant genetic resources across borders has benefited almost all countries around the world. And the best example is wheat, rice, and maize. We all know that wheat is originated from Mediterranean region and rice from Indian subcontinent and maize from Mexico. These are dispersed throughout the globe, and then now they are feeding the entire world, and this reflects the importance of sharing and exchange of germplasm. And we had a wide array of fruit crops in our fruit basket. Mango, banana, and citrus, though originated from India, are now relished by all over the world, and in turn, we are relishing all types of fruits in our country. So we can say that the 7 colors of rainbow in our fruit basket is due to sharing and exchange of germplasm. Thus, germplasm exchange is the way towards global food and nutritional security. Though exchange activity has been taking place, but systematic efforts on exchange of germplasm started with the establishment of Plant Introduction Division in 1946, which led to, and later on led to the establishment of National Bureau of Plant Genetic Resources, and this NBPGR is serving as a single window system for taking place of exchange of plant genetic resources for research use. So in terms of exchange, it is a give and take activity, sharing and getting the germplasm from other countries. So if a new introduce a new germplasm from foreign countries, it is called import. And if we are sharing germplasm, Indian germplasm to other country, it is called export. And if the sharing occurs within our national boundaries, it is called domestic supply. So NVPGR works with a network of its 10 regional stations and 59 national active germplasm sites and other international collaborators and other national stakeholders. And together, we all together work for sharing and exchange of germplasm for research use. So till date, exchange of NBPGR has facilitated exchange of germplasm from over 105 countries and international agriculture research centers. And every year, over 20,000 accessions of germplasm are received for utilization throughout the country. And more than 5 lakh accessions are being supplied to different indenters for use in national programs. So now we'll see how to import germplasm for research use. So first and most important thing is you have to identify the source of availability of germplasm. So this is like a overall activity I have tried to put in one slide. So first you identify what is the source of availability. Then there are two, like there could be two things. If you have to import the germplasm in bulk quantity for commercial purpose, then you'll have to contact Department of Plant and Directorate of Plant Protection and Quarantine Storage at Faridabad. But if you have to import germplasm in small quantity for research purpose, then NBPGR is the concerned organization. And then if we have to import transgenic, then first requirement is permission from Review Committee on Genetic Manipulation set up by Department of Biotechnology. If it is a non-transgenic, then you need to submit application to NBPGR, and the application is Plant Quarantine 08 with prescribed fees and relevant documents. After scrutiny of application, we issue import permit, and the permit is being sent to the indentor for further they can send it to supplier, or directly we can send it to supplier. Once the material is received, we here process for custom clearance, and then material the consignment is released and submitted to Plant Quarantine Division for quarantine inspection. And once the material is found free from insect pests and diseases, then the material is again sent back to our exchange division where EC number is assigned, and then the material is dispatched to indenter. So this is the complete process. And if it is a public sector, then other documents are required. But if it is a private company, then they need to submit certificate of research and development recognized by Department of Scientific and Industrial Research. So now we'll see all the steps in detail. So first you have to identify the source of availability of germplasm. How we'll identify? Through literature survey. So you can do it by your side, or sorry, or otherwise you can ask us to facilitate, like we'll search the source from where, in which gene bank the material is available. You just identify this germplasm you want to import, then we'll search in which gene bank this material is available. And then and there are so many gene banks throughout the world which are sharing germplasm, but the most user-friendly is Germplasm Resource Information Green Database. Here you can search your accession, and then you can send the request to us, or otherwise directly you can apply for import permit mentioning these germplasm are available in USDA gene bank. So once you have the trait or germplasm of your interest, then you should know that there are two requirements. One is import permit, which is issued by NBPGR, and then there is second is phytosanitary certificate. This will be issued by the exporting country. So this phytosanitary certificate is a document regarding the health status of consignment, and it is issued by government official from the source country in the prescribed format of Food and Agriculture Organization. So it should mention about the additional declaration from freedom from specific pests and diseases as specified, or that the pests specified do not occur in the country or state of origin as supported by documentary evidence thereof. Additional declaration means they should give the statement that the planting material is free from the specific pests which are not occurring in our country. So that's why we need an additional declaration from the exporting country. And specific pests for the crops are mentioned in Schedule 5 and 6 of Plant Quarantine Order. So you can visit there and you can see this thing. But all these exercises you need not to do. These all exercises are being done by our side at NBPGR before giving any import permit. And then we'll mention on the import permit that this additional declaration we need in the phytosanitary certificate. Then the exporter will give all this declaration on the phytosanitary certificate. So every consignment should be accompanied by a phytosanitary certificate in original. Then before applying for any import permit, we should know that certain plant species are prohibited from import. So like some plant materials are there which they are prohibited to import from certain countries, like banana cannot be imported from Central and South America, Hawaii and Philippines due to incidence of pests such as Moko wilt and Cameroon marbling. And then in date form cannot be imported from Algeria and Morocco. Chestnut cannot be imported from North America. So first, if you have to import banana, you just check from which country you want to introduce that material. That is very important for quarantine point of view. Then oil palm cannot be imported from Philippines and Guam. And rubber cannot be imported from tropical America. You can see your crop. The whole list is mentioned in plant quarantine order. So you need to look first these things. Then there are certain species where import is permitted import is permitted, but this is there's some problem in the slide.
Yes, ma'am. मैम कुछ दिक्कत है मैम? मैम आप अनम्यूट करिए मैम, आपका ना वॉइस इज नॉट कमिंग. यस मैम. Okay ma'am. Okay.
Slide is moving now. So there are in schedule 5 there are some list of plant material where restricted import is permissible only with the recommendation of authorized institutions. So if banana has to import, then we need the recommendation of Director NRC Banana, and then for citrus, Central Citrus Research Institute, recommendation is needed. For potato, we need approval from CPRI. So likewise, this is not the complete list. Okay. This is not the complete list, but before applying for import permit, you should see that from where for which crop we need a recommendation from a specific crop-based institute. So after that, you can apply for import permit, and this is about import permit. As per clause 6.2 of plant quarantine order, Director NBPGR is authorized to issue import permit, and any researcher with valid R&D certificate recognized by Department of Scientific and Industrial Research can apply for an import permit. And there is prescribed fees for import permit. For non-transgenic, it is 200, and for transgenic, it is 350 for public sector. And the quantity is also specified how much quantity can be imported, maximum quantity. So we'll show it in subsequent slides. And the import permit is valid only for 6 months, and with special justification, it can be extended up to 1 year. This is the fees, and as I have told you, it is available at our website. And then we charge handling and cargo clearance charges. This is per consignment. If you have to import, suppose 50 accessions of cucumber, suppose, then 2,000 rupees you have to pay. And if you have to import 2 accessions, then also it is not per sample, but it is per consignment. So 2,000 for public sector and 4,000 for private sector. Then we charge quarantine processing and clearance fees. This is per sample, 150 rupees per sample, and for vegetative propagule, it is 250 rupees. So these are the charges which the indentor has to pay. And this is the maximum quantity. If it is a large seeded crop, maximum quantity permissible is up to 1 kg. And if it is rice, wheat, and barley, then only 200 grams are permitted. And for very small, it is up to 25 grams. And in case of rooted cuttings, it is up to 25 numbers. The maximum limit is 25. And in case of vegetative propagules like corms or anything you have to import, then up to 15 numbers. This I have listed some of the sources from where you can import fruit genetic resources. So now coming to how to fill PQ08 form. You need to visit our website. There on the, it is mentioned here in the left side, germplasm import. If you go here, then you can see request for pro forma for import of germplasm is available. That is PQ08 form. So you can fill that form. This is PQ08 form. So you need to fill this form. Like we have to name and address of the applicant and R&D. Here, like description of the material to be imported, that is important, like common name and botanical name. And then you need to fill biological status of the material, whether it is a germplasm or a variety or hybrid. If it is a hybrid, then you have to mention its parentage, if it is known. Then the form of material, like seed or tubers or in vitro culture, you have to mention here. And then if you know the actual origin of material, sometimes what happens, Indian origin material, we used to import from USDA. So here we should mention the origin of the material. And then whether it is transgenic or not, here we have to mention. And then here comes the address of the organization from where you want to import the material. So here detailed name and address of the organization. And then number of samples to be imported, suppose 50 accessions. Then you mention 50, and quantity to be imported. If like you have to import 25, in case of horticultural crops, when cuttings are imported, then number of samples is one cutting is one number of sample. So you have to fill like that. And then if you have come to know about this material through a reference, you should mention here. And then if it is shared with GenBank, yes or no, in yes or no, you have to reply here. And then expected date of arrival in India, like if you are applying in February, then you can give by like April, after 2 months, so that it takes at least 1 or 2 months in processing. So you can say 2 months, April. And mode of shipment is air freight. And here you have to give some declaration that the material has no commercial value and it can be shared freely for research purpose. And it does not contain any terminator gene and the exclusively for research purpose. So this declaration they have to give. And for private seed companies, if they have to fill this PQ08 form along with R&D certificate from DSIR. You can also apply online. Here, you can visit this site and then you can log in. If you are a new user, then you can register here, give your details and register, and then user ID and password will be sent to your mail. And then you can fill that form online. And here, like you go to request for germplasm and you can fill the form online. Then you can also make changes in your profile, like that you can change your password. So now we will see how we can fill it, how it looks online application. So here I'll open my account, username. If you have created a username and password, then you can log in. And then here you can see here request for germplasm, view status, manage profile, change password, everything is there. And so you go to request for germplasm. So here you will fill all the requests. As I told you earlier, like here R&D means NBPGR. So you suppose here you have to fill some crop. So everything, all the fields you fill, and then you click to add button. Then it will add it. Okay, all the records. And then you fill all these things and
ma'am, just you click on your screen and
I have clicked.
Is it coming? Arrow, down arrow, ma'am. Yes.
Now it's coming?
No, ma'am, it's not moving.
Okay. Now it's not moving. Okay, some problem, I think. It's not moving.
चलो आगे से कंटिन्यू कर दो। यहाँ से जूम करो जूम। उसको कर दो अच्छी तरह।
Now it's visible. My
yes, slide is visible, but my
okay.
आगे बढ़ा नहीं।
Okay.
Okay.
Okay, no problem. Okay, then like online application, you can, it is very easy, very user friendly. So once you submit that PQ08 form, we process the request at our end, and he is Mr. Satyapal Singh. He is looking after issuance of import permit. He will process your request, and then import permit will be issued. Slide is moving.
Yes.
Okay.
and slides are moving. Yes.
Okay. Then the import permit is issued in triplicate. So this is yellow color is our official copy, and then we'll send these 2 import permits to the indenter. And here you can see that additional declaration are mentioned here. Voice. Okay. So here additional declarations are mentioned here. Is it okay now? Am I audible?
Yes.
Okay. So on the import permit, every or additional declarations are mentioned. So all the exercises we will do, only you have to apply for import permit, and all these exercises are being done at NVPGR. Once we send the import permit to the supplier, they will send the material along with phytosanitary certificate. And here, like Mr. Dr. Surinder Singh is looking after custom clearance and release of consignment. He is our technical officer. So he will send all the documents for custom release of the consignment, and then the materials come like this at exchange unit. And then this material is after official records and all. This is submitted to plant quarantine division for quarantine inspection. Once it is through from quarantine inspection, the material accessioning of the material is done. He is Mr. PC Binda, who is looking after accessioning of material, like means allotment of assignment of EC number. Then the material is sent to the indenter after receiving handling and quarantine fees. So in this form, like we used to get material, particularly in horticultural crops. And that's not the end of the story. We seek your feedback that like whether the material imported is established or not. So the indenter used to send the feedback to us, like when we facilitated the import of pomegranate varieties from France, the material gets established very well. Then the material introduced from USA also got established. And then this is for citrus. And we facilitated the introduction of a new species called monk fruit on the request of IHPT Palampur. And in 2018, we introduced this material, and now the material got established very well, and now it is under the farmer's field. Hing also we introduced on the request of IHPT Palampur, which got established very well at IHPT. And this is about amaranth. So still we have some challenges in sharing of germplasm beyond boundaries. In case of, particularly in case of horticultural crops, the planting material is perishable in nature. So while transportation, sometimes material get destroyed. So that is the main problem. And sometimes what happens when a new material comes to a new environment, then due to maladaptation, the material could not get established. That causes challenges in utilization. And sometimes the material gets very well adapted. Then also sometimes it creates a problem because the monoculture restricts the genetic diversity that we can see particularly in temperate fruit crops like Santa Rosa plum. It is dominating. So sometimes it leads to monoculture also. And nevertheless, introduction of any new germplasm is often subjected to introduction of invasive pests and diseases. So we need to take very special care while importing any material, and strict quarantine inspections are being done. That's why to avoid such problem. And we have a long list of new pests and diseases which are introduced in India with new germplasm. So this is some of the list in fruit crops. So Some important points to remember is the consignment must always be addressed to the director and BPGR New Delhi. Sometimes what happens, they send the material in the name of indentor, then there's a very it is very difficult to release the material from custom clearance. So this should be like whenever you send a letter to the supplier, you please mention that it should be always be addressed to director and BPGR. And the supplier should be instructed to send 2 copies of import permit and other relevant documents with the consignment for custom clearance. The seed should not be treated with any chemical. And NBPGR may retain up to 5% of imported seeds for future reference. And if the material is released with undertaking, the material should be grown under post-entry quarantine facility of the Indenting Institute, and always maintain original national identity or is a number for obvious national and international obligations of access and benefit sharing, and acknowledge the receipt of material and send feedback information on the performance or utilization of the material. Now, procedure for export of germplasm, Dr. S.K. Radha will take over. Sir, please.
Thank you, ma'am. Thank you very much.
Thank you, madam. Good afternoon to all the participants.
Yes, sir.
And Dr. Ravi.
Yes, sir. Good afternoon, sir.
For the session.
Yes, sir.
I hope the participants are not sleeping.
Yes, sir.
After lunch.
Yes, sir.
They might feeling sleepy.
No, sir.
No.
No, sir.
Okay. If 2-3 persons are saying yes sir, no sir, it means they are in active mode. It's good.
Yes.
Yes, sir.
Yes.
Okay,
Yes,
now
sir.
confident.
Yes,
Many are there. Good. This is one of the drawbacks of online classes, online presentations. If it would have been an offline presentation, I can see all of you and pinpoint who is feeling bored or who is sleeping or who is actively participating in this presentation.
Yes.
So, okay. We have to do all these things. Now, as we know that NBPGR is dealing with 3 things: import, export, and domestic supply. These are 3 mandates of National Bureau of Plant Genetic Resources. Import or introduction of plant genetic resources from abroad has been well explained by Dr. Pragya. Now, we will be taking up export of PGR. Before that, why do we need import and export of PGR? It is because it creates diversity in the existing germplasm. The introduction of new germplasm from all other countries is required by the plant breeders for wide genetic base in that particular crop, which leads to elite lines and new plant varieties. As a human psychology, we all need diversity, different colors, different taste. And similarly, in breeding also, we need diversity in genetic stock so that the required traits can be included in the food crops. Now it is one example I can give you. As we know, tomato and apple, these are not of Indian origin crops. Both the crops has been introduced long back from abroad by Britishers, by Mughals, by the travelers. Many crops and species has been introduced. And the recent example we can give of hing, which has been introduced 2-3 years back and is doing well in Himalayan belt at IHBT Palampur. And some 2-3 decades back, kiwi was introduced from Australia, from China, and now kiwi is a well-known crop in Himachal Pradesh, Uttarakhand, and parts of Jammu and Kashmir. In Delhi, we know kiwi fruit, but most of people do not consume it due to lack of awareness or lack of awareness of the taste. But it is introduced crop, and the vitamin C content of kiwi is more than amla and the citrus fruits. So in nutshell, I want to convey you that introduction of germplasm is important to widen the diversity in that particular species. In the same way as we need introduction, the other countries also need introduction in their country, in their for their research purposes. For that, we need to export the germplasm. Before 1992, export of germplasm or import of germplasm was an easy task. But after that, many rules are in existence, many acts are in existence that we have to follow. And a convention was held long back in 92. The aim was to provide conservation of biological diversity and sustainable use of its components. And the third point was fair and equitable sharing of benefits. Under the provisions of that CBD, our country, our government has enacted the legislation that is called Biological Diversity Act, known as BDA 2002 commonly, and BDR 2004. The rules came into existence on 1st October 2003, and the enforcement of these provisions are known as BDA and BDR 2004. Now, these access to all biological resources of India, these are governed by these two BD Act and BD rule for all research purposes. Now, according to Section 1, this act may be called the Biological Diversity Act 2002, and it extends to the whole of the India in our territory. These are some sections and rules. We need not to worry about these. Now, what is biological resource? All types of plants, animals, microorganisms, or parts thereof, their genetic material and byproducts. Byproducts does not include the value-added products. These all are considered as biological resources as per these rules. And what is biosurvey and bioutilization? It is the collection of the species, subspecies, their genes, their components, and extracts of those biological resources for any purpose and excludes characterization. Now, as per subsection 2, the persons who shall be required to take the approval of NBA under subsection 1 are the following persons. One who is not a citizen of India. Then, if a citizen of India who is non-resident and as defined in clause 30 of section 2 of the Income Tax Act 1961. And clause C says that a corporate body, association or organization not incorporated or registered in India or registered, if it is registered in India under any law for the time being in force having non-Indian participation in its share capital or management. These are some technical terms which are to be taken care while indenting a germplasm or exporting a germplasm to other countries. Now, section 4 says that results of research not to be transferred to certain persons without approval of NBA. National Biodiversity Authority, the headquarters are located at Chennai. And no one is authorized to transfer the results of any research relating to any biological resources occurring in or obtained from India for monetary purpose. Means no commercial utilization of that germplasm or that seed should be allowed to any person, to any corporate or any company who is not a citizen of India. And all these things are governed by NBA, headquarters at Chennai. In this, we have to take care that the transfer does not include the publication of research papers or publication of books, presentation of information in seminars, workshops, symposiums, like that. I hope it is clear to all.
Yes, sir.
If any person in between also. Because this is not a one-way lecture. As we know, germplasm is exchange of plant genetic resources. So information and this learning also, it is both ways. I want to learn from you people and you are taking information from me. So you can ask any question whenever it arises to your mind. Now these are 4 categories of germplasm that can be exported. Number 1 is export of crops which are annexed in ITPGRFA. What is ITPGRFA? It is International Treaty on Plant Genetic Resources for Food and Agriculture. These are FAO designated crops which can be exported across the countries which are signatory to that treaty. Second category is the germplasm can be exported under collaborative research projects between 2 institutions, between 2 governments, or like if memorandum of understanding is signed, any research project is running in collaboration. So under that project, we can export the germplasm. Third category is if any Indian researcher or government institution, a university or ICR institute or any other university wants to send the germplasm for non-commercial research or research for emergency purposes other than collaborative research projects. This is the third category in the way we can export the germplasm. The fourth category is with the persons or the request of the germplasm which are not covered in category number 1, 2, and 3. That is access by non-Indian entity, not covered under ITPGRFA, not covered under CRP. CRP means collaborative research projects and non-commercial research as per section 3.2 of Biodiversity Act 2002. So these 4 categories are summarized here. This can be understood in this way, like at the 4th point here, we can see the request for Annex 1 crops designated by ITPGRFA under FAO. These are ICRISAT mandated crops. These can be exported. The applicant is directed to contact DACFW, the National Focal Point Supporting Documents. The recommendations of the director, project coordinator, project director concerned are taken for that particular crop, and the concerned PCPD shall approve. After that, the next step, verification of the FAO designated status by NBPGR in case of ICRISAT FAO designated crops. Then after this step, approval of DAC is taken, and after its approval, issuance of phytosanitary certificate and dispatch of the material. Now, who is authorized to issue the phytosanitary certificate? In export cases, Director NBPGR is authorized to issue this phytosanitary certificate after proper quarantine at Division of Plant Quarantine, NBPGR, New Delhi. Another category is request under collaborative research projects conforming to policy guidelines of central government under section 5 of BD Act 2002. This request is facilitated by ICR and BPGR. The recommendation of the concerned director, PC or PD, and director ICR and BPGR along with the checklist prepared for this purpose. It means a committee has been constituted. In that committee, ICR director and BPGR is always a member to that, and the concerned PC, PD or director of the crop which is to be exported. For example, if a vegetable crop is to be exported, then Director IVR Varanasi. If a fruit crop or a fruit crop is to be exported, then IIHR Bangalore or CITS Srinagar or Lucknow. These are institutes which are to from where the crop is to be exported, and the concerned director is the member of that committee. These PDPCs, the crops they maintain are also known as NAGs. I hope you all may be knowing about the NAGs. NAGs means National Active Germplasm Sites. These National Active Germplasm Sites, they have three types of functions. Means when a crop is introduced to the country, then a part of that material is to be multiplied and maintained by the NAGs. Then if any material is to be exported outside, then these NAGs are supposed to provide the material which is to be exported. For example, some variety is there or some specific trade specific germplasm is there. Then third function is that when a genetic stock is registered at any PGR, the germplasm, particularly the vegetative crops, are to be submitted to these NAGs and a letter is to be signed and applied at any PGR. Now, this is the procedure for export of plant genetic resources for research under collaborative research projects. This is section 5 of BDA 2002 and in an interface meeting at NBA, DARE, ICR and PGR. These are all parties to it. A committee has been constituted to scrutinize the cases. This I have already explained to you. The following documents are required. Request letter from the indenter. Indenter means outside the country from where the request has received. A proper request letter is required from there and import permit from that applicant or from that country. As Dr. Pragya has already explained to you, import permit is generated at NVPGR to introduce the germplasm from outside the country. In the same way, import permit is required from that country for the germplasm to be exported from India to outside. I hope this sentence is clear to all of you. Any query here?
Sir, no.
No, sir. No.
Then copy of MOU, Memorandum of Understanding or bilateral agreement signed by both the parties. Both the parties means both the institutions or both the countries. That copy is required along with the case. List of material to be supplied or exported outside. Then material transfer agreement signed by both the parties. Undertaking by Indian and foreign collaborators, both PIs, for compliance of Biodiversity Act. These documents are required along with the case. After that, the committee constituted, all the members scrutinize the cases for export of the germplasm. Now the export is completed. This is inland supply of plant germplasm or domestic supply. Inland supply means supply of plant germplasm within the country. Now, what are the forms required? What is the procedure to supply the germplasm within the country? The first and foremost requirement of the supply of the germplasm in the country is the indentor, the breeder or the student who wants the germplasm should be very much clear in its goals or targets or in his research. He must be clear about the mandate of his experiments, what he or she has to do. Because sometimes we receive the request of the germplasm in a vague manner. I will tell you afterwards. Now this is the procedure for indenting from NPGR/NAGs. All the requests for supply of germplasm, either stored in LTS, long-term storage, medium-term storage in the National Gene Bank of NVPGR, or maintained by NVPGR in its field gene bank or at its regional stations, or maintained at NAGS, National Active Germplasm Sites. All requests should be sent to Director NVPGR, Pusa Campus, New Delhi. It means NVPGR plays a role of single window system to supply the germplasm for research purposes to the indenters. Now this material transfer agreement, MTA form and JAX-01 form, these two forms are required to get the germplasm within the country. And both these forms are available on NPR website. The website address is given here. You can ask these forms from NPR directly or email also. You can download from website also. But these two forms are mandatory requirement to get any type of germplasm. Here, I would like to request all the participants who are attending this, this lecture, only one crop is to be requested at a time. In one MTA, one crop is to be written. Sometimes some people, they write 3, 4, 5 or 7 crops in the same MTA. It creates confusion at NUPGR because we do not have all the germplasm at NUPGR, or we do not have all the seed material in exchange unit. We have to manage the seed from our National Gene Bank, from our 10 regional stations, and from National Active Germplasm sites. So if 3 or 4 crops are given in one MTA, then, for example, one cucumber is required, one wheat is required, and one mustard is required. Then what will happen? Cucumber will be given immediately from NUPGR, and the case will be filed in supplied cases. In the same case, mustard and wheat are there, and the material is not yet sent to the indentor. Then the case is filed, and it is then could not be followed properly. So in one MTA and JAX01 at a time, one crop, you can write more number of accessions, and more number does not mean in hundreds or thousands. It means only 40 to 50 accessions or 60 accessions in which experiment can be conducted. Sometimes some of our indenters, I don't want to name here because we know all the names and all the institutes' name, who write 300 accessions of this and this crop, 500 accessions of this and this crop. So this should be taken care. Then when the material is received by the indentor, after 2 years of their experiment or 2 years of the receipt of material with the indentor, NUPGR needs the feedback information from the breeder. Because in 2 years or 3 years, the breeder can screen the germplasm, can utilize the germplasm in any form, and after utilization, the feedback information is required. It is required for the purpose so that NUPGR can serve you in a better way. Because without feedback information, we are unable to know what was the wealth of that germplasm, what was the importance of that germplasm. For that purpose, we depend on the breeders or on the indenters who are utilizing that germplasm. Second point which I want to emphasize here is that sufficient quantity of multiplied seed must be sent to Director NUPGR New Delhi. Why the seed is to be sent back? Because NUPGR is giving, providing this seed material, supplying the seed material without any charges. It is free of cost. But as we know, in this era, nothing is free of cost. That cost is to be incurred by someone at somewhere in some terms. Because free of cost, not even the oxygen in air is free of cost. That also costs us a lot. So how we can think that the germplasm, which is precious, the germplasm which has been introduced from outside with number of PQ08 forms and number of import permits, then so and so orders. After so many ifs and buts, the germplasm comes to India. And also, not only introduction, the second source of our germplasm is explorations. NUPGR is involved in explorations around the year. We have a separate division of germplasm exploration and germplasm collection at NVPGR. And that exploration division, in collaboration with the different state agricultural universities and in collaboration with ICR institutes, is conducting continuous explorations across the country from Kashmir to Kanyakumari round the year. Round the year means depending on the harvesting time of that particular crop. Round the year does not mean that every time the explorers are on the traveling roads. That all issues are taken care, which crop is being harvested or being matured at what time of the year. According to that, the explorations are being planned along with the collaborators. And as you all know that NPGR has 10 regional stations, and all those 10 regional stations are in different eco zones of our country. Like to capture the diversity in temperate zones, we have a regional station in Kashmir, then Shimla, and in Uttarakhand at Bhawali. These three are for Western Himalayan zone diversity. Then for arid crops, we have a regional station at Jodhpur. Then down in Maharashtra at Akola. Then for diversity of Western Ghats, we have a regional station at Thrissur. KU people are attending this training. So Kerala Agriculture University in its campus only, NPGR regional station Thrissur is located. Then one regional station is at Ranchi, and for northeastern crops, one regional station is functioning at Shillong, Barapani. So what I want to convey you that after number of explorations, and exploration does not mean ki we collected the seed and put up in the National Gene Bank. The seed, sometimes 50 or 70 seeds or 100 seeds are being collected. They are to be multiplied at NPGR regional stations and New Delhi also. After multiplication, the required number of seeds are deposited in the National Gene Bank. For what purpose? To serve the breeders. The seed is taken out from the gene bank and given to the request which I will receive through MTA and JAX01. My second point in this slide. This MTA and JAX01 are not mere forms. To supply the germplasm on the basis of these two forms, we have to go follow the procedures of introduction, import of germplasm, and follow the procedures of exploration of germplasm. So the third point is sufficient quantity of multiplied seed must be sent to director and NVPGR. Why it is being emphasized here? Because the breeders, after utilization of the germplasm, when they have completed their research work or their experiments, they are left with certain amount of seed, which is not of much use for them. So they can send that seed to NVPGR. And we at NVPGR take care of that seed which has been sent by you people for further supply to the future inventors. Now sufficient quantity means for self-pollinated crops, it is 2000 seeds per accession. And for cross-pollinated crops, it is 4000 number of seeds per accession. Now this is the representation of the same in land supply. The indentor may be from public sector or from private sector. From public sector, the indentor needs to submit an application in JAX01 and MTA form. Both I have already explained to you. Then after we receive the completely filled JAX01 and MTA form, then we scrutinize the applications at NVPGR. After scrutiny, the request is sent to NAGS or NVPGR headquarter or NVPGR regional stations. I have already explained to you. And after sending the request, the seeds, the germplasm is directly sent to the indentor with the information to headquarters. On the other hand, the private indenters, if they are fully Indian, they have to submit the R&D certificates, Research and Development Certificate of DSIR, Department of Scientific and Industrial Research. This certificate is given to all those private companies who are fully Indian and have registered themselves at DSIR. Second paper is undertaking of section 3.2 of BDA 2002 on ₹50 stamp paper. These two papers are required along with JAX 01 and MTA to a private company or to a person who is not in public sector. Then in private only, the other option is if a person is non-Indian under section 3.2, then that person or that company or that institution or that organization who does not fall under section 3.2 has to take permission from National Biodiversity Authority located at Chennai. And they have their different forms. The forms are to be filled up and submitted to NBA. After that, they will decide whether the seed is to be shared with that company or not. When their decision is affirmative, when their decision comes, seed is to be supplied, after that, the indentor has to follow the same procedure of a private company. Means, after permission from NBA, the company has to submit the DSIR certificate and undertaking ₹50 stamp paper. And then follow the same procedure. JAX 01 MTA form, and after all these documents, scrutiny of the application will be done at NVPGR. Then, if the applicant founds perfectly well, signatures at all places. I will explain you about the signatures in my coming slides. And after proper scrutiny, the request is sent to the NVPGR regional stations or NAGS, National Active Germplasm Sites, with a request to send the germplasm directly to the indentor for their research purposes. This is clear to the participants? All are sleeping?
Yes, sir. No.
Only Dr. Ravi is responding.
Yes.
Dr. Ravi has to manage this lecture and he has to coordinate this lecture. So he is responding earliest.
Yes, sir. Yes,
No, sir. No, sir.
Yes.
Sir,
Now
everyone
I'm
is
happy.
listening, sir. Yes.
Now I'm happy.
Sir,
Yes.
one thing, can I interrupt, sir? Sir, we are receiving requests from the students to share. Suppose I'm from Directorate of Floriculture. They want some plants of chrysanthemum, tuberose, or any bulbs like that way.
Okay.
For their MSc and PhD. For that also we need to fill MTA and GX1.
Yes. For any one seed, single seed, to be taken from NBPGR or through NBPGR.
No. They want to take from, suppose our institute.
Some students want to take from your institute.
Yes, or a university. You can say, suppose we are having nearby some universities are there.
Okay, it's your decision. It's your purview. आप किसको क्या देते हैं?
अच्छा sir.
But the proper way is the students should sign the MTA and JAX 01 and submit to NPGR, and a copy shall be returned with DFR.
अच्छा.
No, not student, like its guide should sign.
और उसमें ऐसा है कि I'll show you at the signatures place.
Right.
This is format of MTA. This was Dr. T.N. Saha speaking from DFR.
Yes, sir. Yes, sir.
Yes.
Yes, sir.
Okay, Dr. T.N. This is 5-page material transfer agreement for research use within country for public and private entities. So these are rules and regulations. These are the terms and conditions which are to be satisfied while signing this MTA. On the page 3, on the left hand side, it is recipient column, and on the right hand side, it is providers column. So all the indenters have to sign on the recipient side only, left hand side only. Above is recipient, the person, the scientist or person who wants to do the research, and below is the authorized institutional official. The institute director or his head or dean or director research, they have to sign at the second column. lower column of left hand side. And right hand side is meant for Director NVPGR and Officer in Charge Germplasm Exchange and Policy Unit. They are to sign on the right hand side in the provider column. At several times, we request to the indenters not to sign in the provider column. But many a times, particularly from I can remember, but I don't want to name the persons from TNAU and from Shimoga also. Shimoga people are attending this conference. 3-4 people are there. And from PGTSAU, some 7-8 people are attending this training. Telangana State Agriculture University. They type the provider column also at both places, Director and VPGR. Then we have to use that white ink and properly correct these forms. Sometimes we send back the forms, not to sign here, not to stamp here. So all the recipients are requested, all the indenters are requested to sign only in the recipient column. And in the recipient column, in authorized institutional official, a seal of the authority should be there. Maybe any person, director, dean, director research, head of the division, principal of that college, like that. Here I want to say Dr. T.N. Saha and other also, it is for everyone, that if a student wants the material, he or she can sign in the recipient column, but it will be better if it's his guide or the faculty member, he signs in the recipient column and get the signatures of their dean or director in the authorized institutional official place. So this should be taken care. These are definitions of benefit sharing. It means sharing of benefits arising from use of genetic resources. Then germplasm. What is germplasm? It means whole plant, animal, insect, fish, fungi, microbes, or any part and their propagules, including seeds, vegetative parts, tissue culture, embryo, ova, semen, spawn. All this means NBPGR के साथ NBAGR, NBFGR, NBAII. All are included in this germplasm, in the definition of this germplasm. So though we at NBPGR are dealing only with plants, so need not to worry about all those other things. Genetic resources means any genetic material of actual or potential value. Product means genetic resource that incorporates the material provided under the MTA or any of its genetic parts or components that are ready for commercialization. And what is the meaning of commercialization? It means to sell a product or products for monetary benefits in the open market and has corresponding meaning. So this is commercialization. So what earlier Dr. Pragya has also clearly said, and I am again repeating the same, the introduction of germplasm at NPGR is only for research purposes in small quantity. Quantity has already been shown to you depending on the seed size. And in the same way, the seed which is to be sent for domestic supply or inland supply on the basis of MT and JX01, it is also sent in very small quantity, like 2 to 3 grams or 5 grams maximum per accession, not for commercial use. It is only for research purpose only. This is the 5th and final page of that MTA form. These all informations should be written clearly in a readable manner. Type of genetic resources, clearly write here plants. Common name and scientific name of that particular crop which you want to get through NBPGR. Then type of propagales. What is the mode of that germplasm? Number of accessions, details of the accessions if you know. Quantity required per accession or per sample. Purpose, what is the purpose of your research? It is only screening purpose for some particular insect, pest or virus or something else, or some biotic or abiotic stresses, or it is for breeding purpose, it is for evaluation, for characterization, for augmentation. Sometimes no research work is being conducted, only the inventor wants to augment for its institute or for his university, or for multiplication or other purposes. It is to be specified clearly at 7th place, 7th serial number. And enclose a copy of approval of NBA if applicable. This 8th point is required only by the private companies or private organizations, and that too which has to take permission from NBA, which does not fall under section 3.2 of Biodiversity Act 2002. Then here recipient signature and date. Sometimes it is filled half, partially it is filled, and sometimes fully it is filled, but no signatures are there. So you all participants should be aware of this fact that while writing an application, filling an application, the signatures are mandatory requirement. Then only the application counts. Otherwise, anyone can write anything without signatures. The writing has no meaning. Now this is single page JAX-01 form. It is also available on NVPGR website. It is a request to Director NVPGR, as it is clear from this format. Details of seed or planting material required for research. The botanical name of that crop, common name, number of accessions, either EC or IC number. I hope you are well aware by EC or IC. Some lectures might have been given to you from Conservation Division or from someone else. IC means indigenous collection numbers. Indigenous, those accessions, those germplasm which have been collected through exploration programs or augmented through give and take method from within the country. To those material, IC number is allotted by NVPGR. And EC number is exotic collection number. Exotic collection number means it is a number given to that seed material or those germplasm which has been introduced from abroad through same procedure, import permit and so and so and so, like that. तो जो मटेरियल बाहर से आएगा इंपोर्ट होकर उसी को ही EC नंबर देना है. And these IC and EC numbers are unique identity numbers for that germplasm. These are to be quoted in all your publications. These are to be quoted in all your research papers. These are not to be erased at any point of time. By these numbers only we can locate the germplasm in the National Gene Bank also. So these IC and EC numbers are very much important for us also, and for your publications and for your academics also. Because, for example, if you have developed any elite line or any variety or any germplasm which you want to submit in the National Gene Bank, this EC and IC doesn't mean, this IC number doesn't mean that only NVPGR people can get this IC number. You all can get this IC number for your own germplasm, which has been generated at your own research farms, which has been collected by you from any part of the country. So you can submit that germplasm in National Gene Bank in a particular format. I'll show you in my next slide that format by which you can deposit the germplasm and get the IC number. So this IC number is indigenous collection number given to your germplasm which is being deposited in the National Gene Bank. And that is to be quoted in all future correspondence with NVPGR. So because with the help of the IC and EC, we at NVPGR, we means all divisions of NVPGR, all scientific and technical staff of NVPGR, they can very quickly locate the germplasm. Now next is seed quantity per accession per sample. How much seed quantity is required for your experiments? It is to be written here clearly and in a genuine way. Sometimes for tomato and brinjal seed, they write 50 gram each, 100 gram each. So you see, tomato and brinjal, the test weight is very low. The seed size is very small. So in 100 gram, you can transplant a complete hectare land. But if you need to experiment, plan experiment, we need only 1 or 2 gram seed only. Sometimes 20 or 30 number of seeds only. So no need of 100 gram or 50 gram in these solanaceous crops. So we have to be careful. The same thing I was explaining to you in my previous slides, the indenter must know what he wants to introduce, what he wants from within the country through MTA form, because unless until the indenter knows the nature of the crop, he or she will write for brinjal he needs 100 gram seeds, because he will not be knowing what is the nature of the crop, how much test weight is there, how the size of the seed is there. Now purpose, it may be screening, breeding, evaluation, augmentation, multiplication. I have already explained to you. In MTA form also, these purposes are written. You see, at both places, in MTA form also and in JAX-01 form also, the purpose of getting the germplasm is being asked in the forms in a written manner. It does not mean that one column is blank and write something or anything, or don't write anything, it will be okay. No, we have to critically look into the form, and all columns are to be critically and clearly read and are to be filled, keeping in mind the plan of the experiment, the targets of your research, which are to be conducted. So indenter has to be clear in his targets or in his experiments. It is for screening only, or he wants to do breeding for that particular crop, he wants to do the evaluation work for augmentation, or he wants to do seed multiplication. For that purpose only, seed quantity matters. कि उसको किस पर्पस के लिए वो सीड चाहिए, उसके लिए सीड क्वांटिटी मैटर करता है और उसके लिए नंबर ऑफ एक्सेशंस मैटर करते हैं. Now again in the first column or third column, crop name, here only one crop is preferred. You can write 10 crops also, but one crop will be processed at a faster rate, and the supply of the germplasm of a single crop in one request will be sent at the earliest. 10 crops only, the 10 crops are separate. We have to separate those 10 crops, then we have to find the proper regional station or NAGS site for those 10 crops. Then it will take time to process that case. This is the link to get this JAX-01 form from our website, or you can directly go to the website. There also you will find the JAX-01 and MTA forms written clearly to download that form. Now these are steps which are involved for procuring collections. Requests are received and registered as D case. D case means domestic case. Then request is forwarded to the active collections maintained by Division of Germplasm Evaluation at NPGR New Delhi for supply, and NPGR regional stations. This is the first step when we forward your applications. Your means the indenter's applications. The application as first will go to the evaluation division or the regional stations. All the regional stations have separate mandate to multiply and store the seeds in their MTS and in their field gene banks. So all the regional stations, including our evaluation division, they are crop specific. So when we get a request from any indenter, we decide where this request is to be sent. And again here, I'm emphasizing, when all the regional stations, including our evaluation division at headquarters New Delhi, they have separate mandate, they have separate crops to multiply and to supply. For that purpose only, we need one crop only in one MTA form, one crop request per MTA form. If it is meshed, then the case will be meshed. Then next step is NAGS. If the germplasm is not available at our regional stations and at our headquarters in evaluation division in MTS, medium term storage, then we forward the request application to national active germplasm sites. And if the germplasm is not available at NAGs also, then we go to base collections of National Gene Bank. In National Gene Bank, long-term storage is open in very remote cases, and then we supply the germplasm from there. But again, here the point comes, after supplying the germplasm, when the experiments are completed, after 2 or 3 or 4 years of gap, the indentor should feel the responsibility of seed and sending back to the National Gene Bank. Because in National Gene Bank, we not only in LTS, MTS, or in active collections, we do store only 2,000 seeds for self-pollinated crops and 4,000 seeds for cross-pollinated crops. But when NPGR is continuously going and supplying the seeds to the indenters, it will be exhausted. There will be no seed after a certain time. To fulfill that requirement, we are continuously involved in multiplication of the seeds of different crops and accessions. For all vegetable crops, all grain legumes, all cereals, pulses, oilseed crops, we are continuously multiplying the seeds and depositing in MTS and LTS. In that, we need the help of indenters also to multiply the seed and send back to NVPGR. Now, this is the proforma for feedback information. Clearly write the EC number and IC number which has been supplied to the indentor, then crop name, variety, source or country. If it is introduced material, then source or country is to be written, otherwise source from where the seed has been supplied. It may be any place like evaluation division, any of the regional stations, or any of the NAGS or crop-based institute. Because when the letter is sent to the indentor, the name of the source from where the seed is being supplied is clearly written in that letter. After receiving the seed by the indentor takes care of only the seeds, and he may not keep the letters properly in record. So sometimes this problem comes. Purpose of request. What was the purpose of request? Again, the same question comes here. For what purpose the seed is being requested? Material established or not established, or it is maintained or not maintained. It is to be written here. Then seed supplied to National Gene Bank or other repository. Yes or no? Clear feedback is to be given here. Details of the germplasm utilized in experiment or crop improvement programs. Then publication, if any. If there is some publication, some book chapter, research paper, or technical bulletin, or anything else, the reference of that publication should be given here clearly, so that at NPGR we can update our database, we can update our records, and more or less it will help to the future indenters to supply the germplasm. Because if any accession is of some specific trait, or any tolerant or resistant to some abiotic stress or biotic stress, and if some indenter gives this feedback in this proforma, then it will be recorded or kept in records, and this will be supplied to the future indenters for that particular trait. Now, this is a look of the NPGR website where application forms are available. So, guidelines for registration of plant germplasm. So, this registration of plant germplasm, you have already got this lecture, I hope so, till now. Then requisition proforma JAX01 for the supply of seed or planting material for research through NBPGR. Format of passport data sheet for allotment of IC number. For this format, for this form, I was telling you that all the persons who are involved in research, they can collect or they can develop the germplasm at their experimental sites and deposit the seed to the National Gene Bank at NVPGR New Delhi and get the IC number from NVPGR for their germplasm or for their seed which has been supplied to us. These are guidelines for filling application of PPVFRA. These are approved fee structure for import of germplasm material. As Dr. Pragya has already explained to you the fee structure, That fee structure can also be downloaded from NVPGR website at this point. Then guidelines for submission of seed or propagules with national gene banks, guidelines for documentation and conservation of pork varieties, material transfer agreement. This form is required. This is 5 pages form, which I have explained to you just now. This form is required along with JAX01 form for getting these seeds from within the country through NVPGR. Then application pro forma PQ08 for permit for to issue an import permit to introduce the seed or planting material from abroad. So you can get many more application forms from NPR website in this at this place. So thank you very much for patience listening. Any questions or doubts from participants are welcome.
All participants are requested kindly interact with speakers. If you have any doubts or queries.
Okay. I hope participants
Sir, sir, one doubt, sir.
Yes.
Yes, please.
Sir, research सब you वो अब germplasm ले सकते हैं सर आप NBPGR से?
हाँ.
तो जब रिसर्च वर्क के साथ लेने के लिए वो साइन सिग्नेचर किसको इनफ होता सर? गाइड रहना, डीआर रहना, डायरेक्टर रहना सर?
हाँ, इनमें से कोई भी आपका गाइड साइन कर सकता है.
गाइड भी इनफ सर, कॉम्पिटेंट अथॉरिटी?
Yes.
Competent, no. At least head of the division should be the
Madam,
authority. Head
in
of the
research,
division, at least for competent authority, at least head of the division should be there.
Madam, head of the division means nothing but institution head or
Yeah, institution
university
head. Yes, institution head actually.
Institution head is the enough, no, madam?
Yeah.
Okay, madam. Earlier one problem is happened, my issue.
Okay.
That is the competent authority is superior director of research. No, competent authority sometimes guide with regarding to immediate head. That is enough, no, madam?
Yeah, that is enough, I think. The immediate head is enough to sign
First
and
person is the
put
guide
the
and then
rubber
the
seal.
immediate. Sir. Okay, sir. Thank you, sir. Clarifying.
Okay.
Hello, sir.
Anyone? Yes, please.
Hello, sir. This is Dr. Sadashiv.
Yes.
Sir, to get the IC number for any of the plant material, is it need to submit the seeds to NVPGR?
Yeah.
Yes.
How much quantity for cereals, sir?
2,000 seeds for cereals. Cereals are self-pollinated crops. 2,000 number of seeds are to be submitted.
Sir, if it is a new crop or crop it is not cultivated, then in such cases?
New crop means?
For example,
wild
millet. Wild, sir.
Wild.
Yes.
Wild, sir.
Okay.
For example, there is a crop name called Adle millet. It is rarely you can find it in Karnataka, sir, here and there.
Sir, can you tell the botanical name of this millet?
1 minute, sir. Lacryma joba, I think.
The Coix lacrima jobi.
Coix lacrima jobi, sir.
Job steer millet.
Job steer millet.
Sir, for wild you can submit at least 200 seeds, sir.
200.
200 we will get.
A small number, okay, sir. Because rarely here and there you can find one plant. In each plant you will get 30 to 50 seeds.
Okay, sir. Minimum 50 at least we will see. But
50. Okay.
No, sir. Actually, for cryo we will see the 50. But sir, minimum 200 wild species, na.
Okay, sir. Why I asked this question is very recently I found one plant which is growing isolatedly. There is only one plant that is having hardly 50 to 60 seeds. I took last week, sir. Harvested from the plant. You won't find another plant nearby around, I think it is 30 to 50 kilometers.
Okay.
It is a rare crop.
Sir, but coix is.
So in such cases, is there any liability or strict rule for minimum of 100 seeds? Already I harvested the seeds. It is counted 52 seeds are there from each plant.
Okay, sir. But better, sir, it is more than 200, it is better if it is wild.
And then I
species.
have to grow that one, wait for another year. It is a seasonal crop. Next year it will produce the seeds. Then we can think of sending the seeds.
But in general, sir, coix is widely distributed. Maybe in your region, I don't know.
No, sir. I found it in Sagar, and next in Chikmagalur district, Belur Taluka.
Okay, okay.
That's it.
Okay.
I will try, sir. If I get 100 seeds, I will submit the seeds to get the IC number.
Yes. And any other doubts?
No, sir. Thank you, sir. Very informative talk, sir.
Thank you.
Thank you.
Yadav, sir? Hello? हेलो
थिंक उनका रुक गया है कुछ।
हाँ, यादव सर का मैम कुछ
मैं उनको जरा कॉल कराती हूँ। एक बार स्टार्ट करके।
And anyone having doubts, please interact.
Sir, myself, Dr. Dhanavendan, sir.
Yes, yes.
From Central Silk Board.
Yes, yes.
Sir, regarding getting.
Hello, Dr. Ravi, am I audible now?
Yes, sir. Audible, sir.
Actually, some problem occurred in that computer.
Yes, sir. Yes, sir. I understood.
So there was one query in this chat also. Can we get the wild relatives also? Yes, we can request for wild also, and if it is available in our gene bank or at our NAGs, then it will be supplied to the indentor.
Sir, Dr. G. Tangavendan, he is
Dhanavendan, sir. Dhanavendan.
Yes, yes.
Sir, I recently collected that number with NPPGR collaboration only, that in Koloriang district, Arunachal Pradesh. That only passport information enough, sir, for perennial crop like mulberry, or after characterization only we are getting EC number.
No, you want to get the IC number?
Yes, yes, yes.
Yes, passport information is required, and for getting IC numbers, you have to send the rooted plants of mulberry to the concerned institute who is working on mulberry, of the ICR institute, which has been designated NAS for that mulberry. From the director of that institute, the director of that institute will issue a letter to you. If you have received 2 accessions from Dr. Thanavel, and it has been established here. On the basis of that letter, NPGR will issue you the IC number for those accessions.
Okay, sir. Minimum 200, sir?
No. For it is not 200. I'm not saying 200. For example, the requirement of that institute, I think for crop plants, 3 or 4 plants should be, rooted plants should be submitted there.
Okay, sir.
Seed material for all the participants. Kindly here again, the seed material is to be deposited at NPGR New Delhi. And for propagules, for rooted plants, for suckers, like these crops, like onion, garlic, and these sweet potato, for these crops, the germplasm is to be submitted to the concerned director or concerned NAGS, National Active Germplasm Site.
Okay.
Some crop-based institute. Okay. From that director, he will issue the letter. I have received the so and so germplasm in good condition. And then director NPGR will allot the IC numbers to that accession. Okay.
Okay, thank you.
Sir, you are from, sir? Dr. Tangavendan?
Sir, I am CSRC, Central Sericulture Germplasm Resources Center, Central Silk Board, Hosur, Tamil Nadu.
Okay. I think you are also part of NAGS, na?
Yes, sir. We are a NAG center only. It is declared by NPPGR, NAG center only, because recently I told Narendra Nagi and that Subhash Chandra, we recently went.
In that case, Dr.
I collected, sir. That's why I am asking whether after characterization, otherwise only passport data means I will submit immediately. That's why.
Dr. Thanaveden, in that case, your own directory is authorized to issue you that letter.
Sir.
So you need not to send anywhere.
Okay. But that information I should update in online, or I should submit a hard copy to NPPGR.
There is 2 pages form at our website that I just showed in my presentation in the last slide. Download that and you write the information in that and send through email or send through hard copy to director LPGR.
Okay, thank you, sir.
Along with the recommendation letter of your director.
Okay,
And
sir.
these has been collected and established in our field gene bank.
Okay, sir. Okay, sir.
Like that.
Okay, okay, thank you, sir.
And Dr. Thangavengan,
Sir.
for your kind information, we are sending all the forms, all the
Sir.
essential forms of NBPGR, we are sending to by email to all
Okay.
participants.
Sir.
And meanwhile, we will send publications also. NBPGR is having a free publications.
Yes.
It is available in website also. And Yadav sir also showed the forms, all forms.
Yes.
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 And please, anyone having doubts?
Okay, thank you, sir.
Thank you. I hope all doubts are solved, sir. And meanwhile, in our sessions also it will come, sir. In the first day also they asked questions about the procedures of germplasm exchange. And now time came to thanks to our speakers. Really, it is my pleasure to explain thanks to Yadav sir for emphasizing the easily procedures of germplasm exchange and what are the methodology and procedures and forms regarding. And second is thanks to Pragya ma'am and how she explained very nice, beautifully she explained what are the procedures of the germplasm exchange and all the methods. I hope viewers also satisfied to these conditions of, and if anyone having doubts, you can ask anytime, or in chat box also you can post. Meanwhile, we are coming. And on the Monday, actually, hello. Meanwhile, Monday, we will start the conservation procedures also, and gene bank management and conservation procedures also. You will get a knowledge how to maintain the field gene banks also. Now, Mulberry will come under non-orthodox field gene bank. We have to maintain that one. And sir, thanks to you, sir, Yadav sir, for delivering a nice lecture, and ma'am also.
Thank you.
Thank you very much, ma'am. And all the viewers, all the trainees are requested. If you have any doubts or inquiries, you can post in chat box also. And tomorrow we have some good presentations are there. That is one is genes and QTLs in crops, methods and applications by Dr. GP Misra.
Dr. Ravi, thank you all of you. Shall I leave now?
Yes, sir. Yes, sir. Yes.
Okay. Thanks to all of you. Thanks to
yes, sir. Yes, sir. Thank you, sir. Thank you. Yes. And tomorrow, another session is also there. That is a PGR informatics by Dr. Sunil Achak. He is a well-known, renowned principal scientist, and he established the PGR portal. Actually, it is very, we can say it is a PGR informatics person, and it is very easily we can access. Before that, conventional method will give you a hard task, how to maintain the database and passport system. But it is very easy now if we go the passport data information. And I request all trainees, don't miss that class. You please kindly attend that one. And hopefully we will get you will get materials also. What are the procedures and all these forms. Meanwhile, time to time, we will send all the materials. And thank you very much for kindly patience for listening all the topics. And next, thank you, Vijay Mandalji, for facilitating the internet facility and a Zoom link and monitor all the programs. And thank you one and all. Thank you very much. Tomorrow 9:30 we will assemble in online. And thank you very much.
Thank you very much.
Thank you.